Sorry for the few minutes of delay. Got everything set up and suddenly my slides started misbehaving, but we are probably ready to go. And so good to see everyone here this morning, and I would like to invite John Finch to start us off with a minute.
One of those is different to the one on the board.
Yes,
Right.
Yeah.
Okay, so as a reflection of this institution's recognition of the deep history and culture of this island, the University of Tasmania wishes to acknowledge the Moanina people, the traditional owners and custodians of the land upon which this campus was built. We acknowledge the contemporary Tasmanian Aboriginal community who have survived invasion and dispossession and continue to maintain their identity, culture and Indigenous rights. We also recognise the value of continuing Aboriginal knowledge and cultural practice, which informs our understandings of history, culture, science and environment. The university's role in the research and education and supporting the development of the Tasmanian community.
Thanks, John. Just a few housekeeping before we start this morning. So as we have the students presenting, it's very important that we try to support them by asking questions to help us to learn more about what they've done in their research. And we'll be streaming the session today via Zoom webinar, and so there'll be a recording that will be available at the end of the day for if you want to kind of watch the presentations, or for the students if you want to share with someone who would want to watch your presentation as well, that will be available, as well as on YouTube by next week. So we have 2 other students who will be presenting today. Their presentation will be considered for the Art Institute of Australia Award, even though that might not happen today, but have it at the back of your mind that your presentation will be judged. And by next week, we'll be able to let you know who is the winner. There are bathrooms located just outside the foyer for those who might want to use the bathroom later on. Ensure that your mobile phones are on silent. And if you need to take an urgent call, you might just want to step out into it and do that and then come back in. For those joining us online, there's supposed to be a QR code which we shared, but suddenly my slides has gone bad. We'll try to get it fixed and see if we have that up again. But for those who are in the room, I think we already have that and we can use that. But alternatively, there are scoring sheets around that you can also use if you're in-house. Photographs will be taken throughout the day. We'll use for social media, for those who are presenting. So I hope that will be fine with you. And so our first presenter this morning is Amal.
Good morning to all of you. My name is Amal, and I'm one of the master's students here at UTAS with the research component. And my research is supervised by Dr. Shaheen Alam, who is currently based at Launceston. And today we'll be talking about some of the farmers' perception and adoption barriers for adopting a technique called reduced tillage. It's part of conservation tillage, and some solutions that we have found out through the studies to support the adoption of that technique. Some of the things we'll be talking about here would be background information about the tillage practices, some objectives, methodology, some results we have got, and the main point of discussion followed by recommendations. Starting with the central problems due to which we have to go for reduced tillage and pick it up as an opportunity to go sustainable is eroding soil mass, soil erosion has been a main problem throughout all Tasmania, and we've also got some figures here saying that Tasmania, although in comparison have less soil erosion than any other states in Australia, but the soil formation rate is 10 times less than soil losing rate, so which is a great number, and we have to look into it as soon as we can. Another problem that we have to discuss here would be waterlogging problems. That figure up there defines most of the Tasmanian soils have a soil volume less porosity, which is less than 4%, which is not usually recommended for most of the soils good for cropping. Another problem that we'll focus is the limited supply of water and expensive inputs and other chemicals. Talking about the solution, just a basic comparison, conventional tillage, maximum manipulation of soil, putting the soil upside down, whereas no-till, which is also part of conservation tillage, but minimal manipulation and buildup of that organic matter is the major outcome. Conservation tillage will focus reduced tillage, where some part of the land is tilled, but most of the part is kept organic, or I would say we leave residues and do some other practices, which we will be talking in a minute. These are some figures that I've got throughout Australia. Mass adoption was there for reduced tillage as well as with no-till, especially no-till. But down the line, we can see reduced tillage has been at a decline phase after 1990, I would say. This is all Australia we're talking about. These are the figures for specifically some part of Australia, not including Northern Territory, but specifically, I pointed out here, Tasmanian farming, where the adoption rate of that reduced till is much less than any other states in Australia, which is around 75%. And you can see all the other states have more than 85%. This was our objectives of the study, 12-week timeline, starting with first 4 weeks, access maximum amount of data that we can, then identify the barriers to the adoption, plus after 4 weeks, we got into interviews with farmers all around the state, and we have gone through some case studies through to the final few weeks, then presenting our data in this presentation as well. Our methodology was pretty much straightforward, not really complicated. We accessed 2 major tools: root cause analysis, gathered information that is important. According to that, then we placed our data and then analyzed it, then reviewed it multiple times. And we were able to build a mind map, which I'll be showing in a second, and also the system analysis, which we use to put core elements, core problems inside. This is the upper circle would be the direct environment, directly involved in pushing those problems, and the last circle, which is indirect environment, which is a key driver, but don't have any direct involvement to the problems that we're talking about. And we got some interviews of around 20 to 25 stakeholders, including farmers here in Tasmania, outside of Tasmania, outside of Australia, some manufacturers, some researchers, including professors as well. What was the results? We can see here a mind map, lower adoption rate being the central problem, branches and sub-branches. One of the major problems that we can say that can be adoption barrier would be the knowledge gap and farmers' perception, with some economic barriers. Same with three-circle diagram, knowledge and training, and perception of yield, farmers' perception of profitability being common in both these results. After doing all those interviews, we were able to collect the data showing us cost of herbicide with being the major adoption barrier to this no tillage, because which we'll be talking about in a minute. But just to be clear, that we'll be talking about 4 common adoption barriers that we have seen throughout the study. We have selected some farmers, including no-till farmers, multiple-till farmers who are practicing cultivation, conventional tillage, and combination farmers who are only practicing no-till, and some of the conventional part as well. So multiple-till farmers pointed out that the major barrier that they have seen throughout their farming system, if they go for no-till or reduced till, would be profitability down, or they will have some soil-specific problems. Combination farmers, they pointed out that and herbicide resistance being a major problem while using these techniques. No-till farmers, whereas pointed out disease control, which is a result of overwintering of spores. When we leave the residue on the ground, they overwinter and create problems then to be removed. It's not easy task for the farmers who are reduced till or no-till. Adoption barriers, that is the major point of discussion. The first one was the common phrase that I have heard from most of the farmers: my farm don't need fixing. They don't need absolute no fixing because we are okay, we have productivity. So why do we have to switch to a new farm? Creating that psychological resistance, and just we don't want it. That's it. And the last thing they mentioned also, the last thing also they mentioned is one problem is nutrient stratification. Top 2 inches of the soil have all the non-moving nutrients like phosphorus. So the roots grow deep and they need access to the nutrients. But if we are using technique like no-till and reduced till, we can't access the plants can't access those non-moving nutrients. One of the things they also mentioned is they have large-scale business. Most of production going for vegetables, not much for the residues to be collected. Not much residues over there, and we actually have to import or buy some of that residue or organic matter to spread throughout the field, which focuses on the initial cost, and they just don't want to do it. Another problem would be soil compaction that they do have in their farm already, due to which they are forced to go for conventional tillage again and again to loosen up soil rather than going for a trial and experiment. Will be a bad investment for them if it doesn't work out. Which brings us to our second problem that we have shown here. This is some numbers that I've got. Herbicides being used among farmers who are using these techniques like no-till and reduced till is 100%. So conventional tillage already uses pretty much, but herbicide usage is pretty high, and we all know it's pretty expensive as well. But farmers are only focusing on the herbicide cost and the labor cost rather than what they can get from it, out of it, which I'll be discussing in a second. One of the other things that they've been focusing on is herbicide resistance, specifically knockdowns. They told us that because of the overwintering of spores, which has been a problem, it actually invites more bugs into our farm, which actually doubles their expenses because they have to use multiple chemicals now to remove that residue they have which have been there, then the weeds, then the insects now they are prone to. So which is like really heavy on the initial cost again. Bringing us to our third problem, lack of machinery. Access to these no-till or reduced-till machinery has been a challenge for the farmers. We don't have really many manufacturers throughout the state who can actually produce no-till machinery. So we do have to get the access from mainland or from outside Australia, which is, again, the initial cost is really high for them. They also told us if it doesn't work out, let's just say we got all the machinery, all the techniques in, all the consultants in for advisory. If it doesn't work out within 2 to 3 years, it's just a bad investment. So we don't want to do it. Finally, limited access support, farmer support, got a number here. They pay advisory much less than any other state, and also psychological residence that our peers are not doing it, why should we? So other farmers who are neighbors to us, they are not doing it, why should we? And also there are no any specific bodies to support that no-till or reduced-till method like we do have in Victoria, the no-till Victoria farmers who actually support this claim and support these methods. Another one is Western Australia, but we don't have any association here in Tasmania who can actually support this technique here. Finally, if a final recommendation would be when you are contracting, you have leased a farm and you have contracts lined up for that farm, you can actually go for easing that contract so that you can practice no-till, because sometimes contract obligation force us not to do that no-till because we then do have to do the conventional tillage because of those contracts. Having a trademark like RSPCA or organic, so that we can push farmers to have that trademark on their products, which can actually give them the incentive to make more of the products out of no-till technique or reduced-till technique, showing them that they will have more profits than the conventional tillage. Time being 1 minute. I'm sorry, I'm saying that. Thank you.
Any questions for Amal? If you're online, you can put your question in the Q&A section of the platform, and your questions will be read. But for those who are in here, do we have anyone with?
So we've seen very strong uptake of no-till on the mainland.
Sorry?
So there's been very strong uptake
Yep.
of no-till on the mainland.
Yeah. It's a very strong uptake, but in comparison,
I haven't asked my question yet. So they don't have a trademark. Wait. So what has been their incentive for the uptake of no-till as opposed to in Tasmania? In Tasmania.
I haven't think it through, but what's their incentive like?
Yeah, why is there such big uptake of no-till on the mainland compared to here?
Well, specifically, one that I mentioned here, they have particular association, like in Victoria or Western Australia, they do have association that supports no-till techniques or reduced-till techniques. But here in Tasmania, we don't have that kind of association who actually supports or spread that knowledge among farmers to practice, and what are the benefits and what are the long-term benefits of these techniques.
So a knowledge gap and support for that knowledge. Thank you.
Yeah.
I just wonder, in relation to the data you've collected here in Tasmania,
Yes.
were you able to differentiate between those cropping, vegetable cropping,
Mhm.
where they might put crops up on bed versus broadacre cropping, cereals and so forth? Were you able to differentiate the views of those growers?
Yes, it's like vegetable growers, most of the farmers who grow actual vegetables, they have some common problems, but they have some separate problems as well. They were actually mentioning to us that they have mentioned to us that like I mentioned, like residue problems. They don't have much for residues here when they are growing any vegetable crops, although in comparison to any grain crop or any pasture-based system, they have a lot of residue to be to go for no-till technique, which is a crucial aspect of no-till technique to use those residues. But yeah, with vegetable farmers, they don't have much of them. They actually have to buy the organic matter, or they will have to buy if they do this tillage technique for the future. So yeah, it's about that cost for property acquisition aspect.
Do you have any online questions? Any more questions?
This is you'll recognise the pattern here, because this is maybe the first silly question of the day. You talk about resistance to change, and I was really curious about one of the key insights that you listed when you were talking about nutrients in the top layer, specifically talking about phosphorus, I think, in the example. And the units that you provided from those key insights was 1 to 2 inches.
Yeah.
It's very old fashioned, isn't it?
Like when we use
Were those your key insights, or was that key insights from literature?
from literature. No, it's one of the farmers that told us that one of the 2 inches of the soil, it actually stays there, and they will create problems, because we talk to farmers like us, Australia, and outside of Tasmania, and they also mentioned that one of the farmers, just not like old farmers, just one of the farmers they mentioned. So I thought it's a good idea to put up in the presentation as well.
Thanks, everyone. Amal will still be here. So if you have any question, you can catch up with him during the morning to provide his answers.
Morning, everyone. I'm Bharath Dinakaran, and I'll be presenting my master's thesis on the Eristalis tenax, novel Tasmanian pollinator, study that I did to mass rear it commercially. Did all of you know what is pollination? Pollination is a basic process through which the pollen grains are transferred from male to female part of a plant. Why pollination is necessary? Because it's the grassroots level of food security, and world's top 100 most crops that we humans consume are totally dependent on these insects for pollination, and the food that we eat on our plates are totally reliant on them. Did you know that 70% of the pollinated crops around the world is through insects, and out of them, the 72% is through honeybees, and the rest, 28% is through the alternative pollinations, that is, be it mechanical pollination, air pollination, or pollination through birds, or any other means of pollination. So this says that we are hugely dependent on honeybees solely for pollination, that to European honeybees. Well, why do we need to study alternate pollinators when we already have honeybees as our basic source of pollination? A key answer to this is we are overly dependent on honeybees, posing a risk to the several industries that are directly or indirectly dependent on food, be it seed industries, be it farmers, or be it different stakeholders in the agriculture production. Also, we know that there was a pest attack in New Zealand, a neighboring country, where the varroa mite was a huge problem for the farmers and bee growers. This pest, not so far that it entered Australia, now it's confined to the New South Wales, but thank God for the biosecurity people. It's confined in New South Wales. We don't have them in Tasmania, but touch wood, we we don't want it to be here. Also, to match the demand and supply. Throughout the year, we can't say that honeybees are active throughout the year, and we have these flies which are Eristalis or the hoverflies, which we can use as alternative to the honeybees in order to pollinate them in the open fields. We have very less knowledge about commercially using them, so this study will dig further into this more. And also, as I said, the world's top 100 most consumed crops by humans is reliant upon the honeybees for pollination. So that's a huge market out there, which is $800 billion worth of market. So pollination is a basic necessity, and the 800 billion market is reliant upon them. Using hoverflies or syrphids for pollination. Syrphids, did you know that syrphids are the second most frequent flower visitors to the honeybees? Well, that's actually an advantage which puts us forward to use this as an alternative pollinator. Second thing, for all those who are scared of insects or honeybees, letting you know that this insect doesn't sting you. It's not allergic. And it delivers as much as equal and efficient pollen grains as the honeybees, and equally efficient. And what if I say that it can be reared in your backyard? People rear them in your household backyards with the kitchen waste or any pungent waste that's organic material with watery solution to easily grow the larvae and provide its maintain its life cycle. Well, when we have all these advantages, there are two sides, there are two edges of the knife where the sharpie edges. We know that this is an efficient pollinator, but it's not present in huge numbers. What can we do to commercially rear them in large quantity and large numbers? Well, we conducted some experiments, and certain key issues involving large number of rearing of this insect is it's very laborious, high input cost, and optimal rearing diet is quite unknown for at the moment. We do know the diet it prefers through this experiment, but there are previous works that has been done. We conducted a series of experiments through cages by using 5 different diets in order to see which was more preferably eaten by the flies, where I used canola, mustard, buckwheat, broad bean, and control. When I say control had a mixture of bee pollen. So when I say bee pollen, how is bee pollen collected is when honeybees are reared in apiaries in a bee box, the cage is prevented with a brush kind of structure where the pollen grains are collected when the honeybee enters into the beehive. And we conducted a series of experiments, and this is how the cage looks like. We bought the adult flies, set them up in the cages with ratios of 25 male to 25 female. And that's the seed purity lab from where we got the adult insects from, the hoverflies, reared them, made sure that we had enough replications, 4 replications per treatment, took regular observations on them, made sure that the flies had enough supply of food through pollen grains, sugar supply, that's sucrose, and water, because these flies don't like to be running out of dehydration. And made sure that the sucrose was updated once in a week in the control, because when I said control, the bee pollen, we gave them enough source of food through additional pollen, sugar, that is sucrose, and water. All the supplies were topped up regularly twice a week. Made sure that the egg counts were recorded, hatching ratios were recorded, the adult mortalities were recorded once in a week or twice a week. And we did run some nitrogen analysis with the help of Central Science Laboratory in the University of Tasmania, which backs us up with a reason why did these flies like a particular diet. And that's how the eggs look like. It's very tiny, it's really tiny. It's just a magnified image. These are the results that I obtained. So when I ran a series of tests with different treatments, so surprisingly, I mean, not any surprise that it preferred bee pollen more than any other treatments that we studied it with. It had more number of eggs laid in the control comparatively to the mustard followed by canola and buckwheat. We didn't find any eggs in the broad bean. The flies clearly disliked the broadbean, probably because of its structure or the flower. And this is the hatching ratios, which is in percentage. We can clearly find again that the flies liked control, that is the bee pollen supplement, more than any other ones, followed by mustard and canola. We couldn't find any hatching ones in the buckwheat or the broadbean. I have a reason to this. When we dig why it happened, well, we have a reason to that is in the Central Science Laboratory, when we tested all these different pollen grains that was collected for the diet, we found that control, that is the bee pollen, had highest nitrogen percentage, and it directly contributed to the egg laying capacity. It's straight away linking that to here. And it's the same with hatching ratios. The nitrogen, the treatment which had nitrogen present more in the pollen grains directly contributed to the highest hatching rate and higher egg laying ratios. And I ran an ANOVA analysis test. I ran an ANOVA analysis to find the significant impact that the pollen grains had to do with the diet that the flies consume. We had a QKHSC test followed by ANOVA, which significantly gave us a value that says it's reliable, and pollen grains had a significant impact on the diet that the flies consume. Well, there are certain conclusions that we can carry as a takeaway lesson from this research. The flies preferred the bee pollen grains, that is the mixture of different pollens, rather than preferring the treatments that we ran with, like mustard or canola or buckwheat or broadbean. Probably it's because the preferred diet had more amount of nitrogen percentage. And the conclusion that I would like to give to it is the eggs that were laid in huge quantity had a direct impact through the nitrogen percentage of the diet it consumed. All the flies that were particularly laying eggs in the control had more number of eggs compared to the flies that were reared with mustard, broadbean, canola, or buckwheat. Well, mustard and canola had a greater impact comparatively to buckwheat and broadbean, where broadbean had no eggs laid by the flies. The bee pollen was the most nitrogen present pollen grains that we had in the experiment, and that triggered its fecundity level of the fly, and the more number of eggs were found in them. What future work can be done in this aspect? Studying this as a base for the future studies of mass rearing of Eristalis tenax, that is the hoverfly, as an alternative pollinator can be deeply studied. Well, this is not the only alternate pollinator that we know. Well, keeping this as a base, we can study other alternative pollinators as well that can be more efficiently used for mass rearing. And in our study, we found that the bee pollen, that is the control, had a much higher nitrogen percentage. If we use this bee pollen with the other treatments that we used in a certain ratio, finding out which ratio of the other treatments could be used in a better way to cost efficiently rear it, it would be a groundbreaking result. And using this study as a base for other rearing methods, which can be cheaply or used as an alternative to the stakeholder engagement by the farmers, and teaching them on how to efficiently use alternative pollinators has to be seen. Also, we can give this as a cheaper alternative to the rearing of honeybees. At present, this is a costlier rearing method, but we can still find out and figure the ways, as in how to cut in the costs and make this an efficient rearing method. Well, thank you so much for patiently listening, and if there's any questions.
Anybody in the room with a question for Bharath? Rhiannon?
I just got a question. Why did you pick those for specific sort of crops to feed them?
Well, they're extremely the performance is really good in the brassicae, and because of the structure of the flower and the structure of the insect, it finds it more easier to consume them. And as we know, the Eristalis is quite similar to the honeybee that instructions, and it's preferably using that as a pathogen mimic. So it's compatible with them, and we tried seeing which can be used in a better cost-efficient manner. Well, there's already works on that area. We just are looking at how to better rear it with cost-efficient method.
Shannon, did you have a question?
I did. It was about, so you said the nitrogen seemed to be really important, and I know nothing about these, so again, might be silly question number 2. Is there a way that you could supplement, like make sure they have more nitrogen?
So why did we analyze? I want to answer to this question by saying, why did we analyze nitrogen first?
Yeah.
The nitrogen, a chain of nitrogen is amino acid.
Yeah.
And amino acids are proteins.
Yeah.
So we tried looking at protein-rich food, which had a significant impact on the egg-laying capacity. So protein, when is consumed by the flies, it's directly involved in its fecundity level. So that's the reason we tried looking into the nitrogen. And
Yeah, I was just wondering if there's some way you could add nitrogen to their diet other than changing the type
as
of
of now,
pollen, but
we just tried it with the crops.
Yeah.
There might be some additional supplement in the future, where we use the bee supplement, right? So that's actually a supplement. It's, you need not stay dependent on the crops forever for the additional supplement. So, but it's a costly one. Right.
I was just wondering about your controls. Did you have a look at the pollen just to potentially identify the species making up the controls, so where that pollen's coming from?
It's just a normal bee pollen that we collect from the shop. It's just normal bee pollen. You don't have to be specifically asking, but we checked the nitrogen percentage that was available in that bee pollen, the content in the control. So it had the highest nitrogen percentage when we analyzed, but we didn't actually know what was the basic constituent of the control, what was the makeup of the control.
How do you know whether the performance of the bee pollen treatment isn't more associated with the flies not having to forage for the pollen themselves rather than the pollen source, i.e. they get the pollen supplied in bulk versus having to find it?
I'm sorry.
No, that's all right. How do you know whether the performance of the bee pollen treatment isn't more associated with the flies not having to forage for the pollen themselves rather than the pollen source, i.e. they get the pollen supplied in bulk versus having to find it?
Can I see the question?
I think it's about landing on a flyer versus collecting it.
No, it's legitimate.
They're not flying for the pollen.
Yes. We are basically concentrating on multiplying or more of eggs here. That is, we want the flies to be more rapid in terms of output. So we're just looking at mass rearing methods, and its foraging behavior is not affected because we tried rearing them in a cage, and the flies had to. So in mustard and canola, we didn't actually supply them with the pollen grains. They had to actually forage for them. Just the plants were kept in the cages. They had to take their own pollen. But in case of control, yes, it was provided as a platter.
Right. Probably because of our time as well. Going.
Can you show them the very first slide that you did? That you didn't actually show.
Oh yeah.
Just quickly, very first one. Keep going, quick. Wasn't there one more before that? The AI one?
Oh yeah.
All right. Russ generated that by AI. That's an AI image that he managed to get, which is amazing.
could you turn the lights down, please? Because you can't see the slides well.
Got popcorn?
Podcast is too now, Shannon, see, it makes me giddy.
Thank you, everyone.
Thanks, Russ.
Our next presenter is Harsheen. Harsheen Kaur, I'll bring her quickly.
You're recorded.
So good morning all. My name is Harsheen Kaur and I'm here to present the research findings on my project Safety and Stability of Plant-Based Milks. I conducted this under the guidance of Professor John Bowman, and I'm thrilled to share my research findings. A bit of a background, plant-based milks are made from sources such as nuts, cereals, legumes, and other types of sources. And they are designed to appear and taste such as cow's milk. The global market of PBMA's plant-based milk alternatives is growing and is projected to land at $28.78 billion by 2030. And there's a staggering milk sales in US especially, which is $344 million. It was in 2021. So why this shift is happening? Because people are moving towards vegan diets and due to increased lactose intolerance, and people are moving towards health conscious lifestyles, which they are considering low calorie diets. So my project is focusing on ensuring that these plant-based milk alternatives are safe to consume. So the problem statement says it investigates the spoilage potential of almond and oat milk compared to bovine milk, especially after opening the UHT packs and storing them at different temperatures, specifically focusing on Bacillus cereus and related strains. So there's a rising demand of plant-based milk alternatives, and there is a risk of spoilage after opening the UHT packs, but there's a lack of data on microbial growth in UHT PBMAs. And the spoilage can cause a lot of food waste, deterioration of quality of milk, and foodborne illnesses. So the aim of our study is to compare the spoilage potential of UHT milks, almond, oat, bovine at 4 degrees Celsius, 8 degrees Celsius, and 20 degrees Celsius with a focus on measuring Bacillus cereus specifically, and to investigate the impact of added sugar on Bacillus cereus proliferation. And also an important aspect of this research was to assess the organoleptic quality changes after opening the pack of these 5 types of milks. So why did I focus on Bacillus cereus? Bacillus cereus is a spore former, and although UHT is designed to kill all bacteria and spores, but there's a risk after opening the UHT pack. And Bacillus cereus is pathogenic, is widespread in nature, is a toxin producer, and has a high risk of causing foodborne illnesses. Given its psychotolerant nature, this was ideal organism to study for this plant-based milk alternatives. My experimental design includes 5 types of milks: almond milk, oat milk, their sweetened and unsweetened versions, cow milk, and the sugar was in the form of added sucrose. We stored these milks at 3 different temperatures, 4, 8, and 20 degrees Celsius. And this study involved microbial analysis, sensory analysis, where 2 replicates of each type of milk was used. And this study utilized the total 5 strains of Bacillus cereus, which were collected and preserved earlier in the research conducted in 2018 by Professor Tom Ross and Zoe Bartlett during the research in Tasmanian dairy farms. So we are grateful for that. Now let's deep dive into the results part. After modeling the growth patterns of Bacillus cereus over time, it was observed that at 8 degrees Celsius and 20 degrees Celsius, almond and oat milk have shown the similar trend as compared to the bovine milk, that the maximum rates are lower as compared to bovine milk, but the lag phase is longer. But when we compare almond milk and oat milk, it was seen that the almond milk has higher proliferation rate for Bacillus cereus over time. Then we conducted statistical analysis, and it was found that there was no statistical significance between results in different types of milks and due to the added sucrose at 4 and 8 degrees Celsius. But at 20 degrees Celsius, there was statistical significant difference between almond and oat milk and oat and bovine milk. And there was also statistical significant difference due to added sucrose in almond milk, but there was no difference in oat milk, sweetened and unsweetened versions. And then it was also observed that there was statistical significant difference between maximum rates of growth at 8 and 20 degrees Celsius. And these results align with another research which was conducted in 2022 by Batula and a researcher in Ireland, which says there was no significant growth at 4 degrees Celsius, but almond milk was supporting the proliferation of Bacillus cereus similar to bovine milk. We also conducted sensory analysis, and this part is ongoing and has reached day 7 only. And it is observed that the overall liking for sweetened products is more as compared to unsweetened ones. And it is also observed that bovine milk has larger acceptability on hedonic scale rating as compared to all other types of PBMAs. Now coming to the attributes, the almond milk have shown that it's exhibited off-white color, but the oat milk was slightly darker. And in contrast, bovine milk was white in color, but almond milk give a nutty prominent flavor in both versions. And there was distinct feature between almond unsweetened and sweetened one, as sweetened one gives more creamier texture, which was liked by the participants. And there was statistically significant difference between all the types of milks like almond, oat, and bovine. And there was significant difference between sweetened and unsweetened versions also, but there was no relation between beverage type and added sucrose level. This part of study is still ongoing, but still it can be concluded that temperature has a significant effect on the acceptability of milks. And storage time also have a significant effect on the acceptability of milks, as the milk stored at 4 degrees Celsius are more preferred and liked on later stage also for the on the hedonic scale. And sucrose level also has significant impact on the beverage liking, as beverages with added sucrose have overall higher acceptability than the unsweetened ones. So this study recommends that there is need to investigate growth patterns of other pathogenic microorganisms like Listeria, E. coli, and Salmonella, etc. And with the more types of PBMAs to be considered, because we studied only 2 types of plant-based milk alternatives, but there are around 150 in European markets. And we are conducting sensory analysis with just 4 participants, but a large number of participants can give more insights about the consumer perceptions. To conclude, although UST is designed to kill all bacteria and spores, including Bacillus cereus, but there's still a risk once pack is opened and left out for extended periods in our homes and at warmer temperatures. So when we did growth modeling, it suggests that PBMA products, especially psychotrophic Bacillus cereus growth, is comparable and has 2 to 6 day lag phase at 8 degrees, while growth is negligible at 4 degrees. So it is recommended to follow the recommendations which packs are saying. And the presence of added sugar in PBMAs was found to influence the Bacillus proliferation only significantly at 20 degrees Celsius. And almond milk supported the greatest proliferation of Bacillus cereus strains, followed by bovine milk, and then oat milk showed least susceptibility. Organoleptic quality assessments demonstrated noticeable changes in sensory attributes under different storage temperatures, and this part of study is still ongoing. And at the end, I want to thanks for all the support and guidance to my supervisor, Professor John Bowman, and to all the TIA faculty members who and all the friends and family who have supported me throughout this journey. Thanks for listening.
Any questions?
Thank you. I'm just curious, why does almond milk support the greatest proliferation over oat milk and bovine milk? Just from a food science content perspective, what's in almonds that drives that growth?
That's a good question, but I might have to look into research more about this at this point of time.
May I comment?
Yes, you can.
Potentially, and I'm not sure, I'm just off the cuff, iron levels, iron as mineral really enhances bacterial growth. So I don't know the iron content difference between the two. It's just a thought.
Gee, thanks for your presentation. Just wanted to clarify, when you did the sensory assessment, was it blind so that the participants didn't know what the milk was? It's just curious to know why they preferred milk, for example, versus almond milk. Could you clarify that?
Not blind.
Was that they didn't know what they were?
Yes.
I think in hindsight, maybe they
Because they-
should have been blind. Is there a
because
reason
they-
why?
Because they check the spoilage based on the control, but the pack was, or the control pack was the fresh one open on that day only, and they have to check the spoilage based on that.
They didn't have to, I'm just saying, it'd be interesting to know whether the participants could actually detect
Yes.
or identify what they were drinking.
It was not
Thanks.
blind.
Thank you.
Thank you.
I also have, I don't know, we could talk forever about the sensory element, but a follow-up to sensory work. We've seen previously that fat content is really important for aroma release. So you've measured and you've normalized for sugar content. Have you controlled for fat content across the different studies?
No, we didn't consider the fat content, although this oat sweetened milk was having more fat content as compared to all of the others. So the liking for this came high as per the participants' scores. But we considered only the sucrose content.
Is it okay if I have one more
Yeah, maybe
follow-up? So, if there's someone in the room who always drinks dairy milk and they are presented with oat milk, it's going to taste like poison. And so, how did you screen for participants in your sensory work?
So the sensory analysis was to understand the consumer perception. We didn't do the screening based on their preference of the milks. It was just a normal screening so that they can understand, and we trained them like these are the tastes and attributes of these different types of milks. And we didn't specifically screen them based on their preference because we want to understand what the actual world is perceiving on these milks.
That's a reason not to have a blind taste, because you know what to expect from the different milks if you've had some training. So that's a good reason not to have a blind tasting.
Thank you.
Good morning, everyone. My name is Kalpana Shahi, a student of Masters of Agriculture and Food Science here in Utah. Today, I will be presenting my work on understanding the susceptibility of leaves at different growth stages to a rust pathogen, Pastures minimum. Throughout this work, I was supervised by Professor Cara Barry from TIA. Thanks to her. Blueberries are native to North America. They were first grown in Australia in 1974 in the Victoria State commercially. Rust pathogen, which was previously called as Phycospora minima and now called as Pastures minimum, was first detected in Australia in 2001. Here in Tasmania, it was first detected in the year 2014, and in the year 2022, it was declared endemic because of its widespread detection in various very regions in Tasmania. Severe rust infection leads to heavy defoliation, which cause ultimate reduction in the fruit. Blueberry rust can be managed by the timely application of fungicides, but there is little information available on at what stage the leaves are more susceptible to this frost pathogen. And at what stage the application of these fungicides can be more effective and efficient. Some of the studies shows that there is a relation between the leafage and the susceptibility pattern. In soybean rose study, it showed that older leaves are more susceptible to rust pathogen compared to the younger. And this was in related with the leaf physiology and the chemistry as well. So our research questions were, is there any variation in susceptibility to frost pathogen with the age of the leaves? What are the biochemical factors in the leaves of blueberry at different growth stages, and what is their connection with the susceptibility? And at last, how could this knowledge be applied to targeted frost management? Our research aim was to investigate the susceptibility of blueberry leaves at different growth stages to the rust pathogen. And our objectives were to investigate the impact of leaf age on the susceptibility to Fixation ester minimum, as well as to quantify the treatment, particularly chlorophyll, and to quantify biochemicals, particularly total soluble sugars and phenols in the leaves at different ages. In the main trial, for the trial, we selected a commercial blueberry orchard in Hume Valley, and we selected legacy cultivar. The first step was to go to the field to check for the emergence of new leaves, because we were conducting the trial in new emerging leaves rather than in the older leaves. So we went to the field, track for the emergence of leaves, tag them. When we get the required amount of tagging, we classify the plant leaves into 3 age groups. The first age group was the oldest leaves group, which were about 4 weeks older. And the second age group was the leaves of age 3 weeks older. And the youngest age group was 3, which was 1 to 2 weeks older. If on the day of bringing the plant back to the glass house, because we were conducting the rust inoculation in the glass house, we did the SPAT reading for the chlorophyll content. We took several leaves of different ages for chlorophyll and phenol analysis, and we got the branches 3 from 1 tree. Altogether, there were 3 plants. And from each plant, we took 3 branches. And from each branches, we took 3 secondary branches having different leaves. The branches that we brought back were kept in the glass house for frost inoculation. It would be more effective if we had like the chance to in the natural environment or in the field. But it is not acceptable for us to introduce the rust pathogen everywhere or to make a great loss. So we just tried doing that with the glass house. So the other process was conducted in the laboratory. It was basically the analysis of biochemicals. This is the lab glass house setup for the inoculation. In the laboratory, we did analysis for the phenols and sugars. For phenols, we first dried the leaves in a freeze dryer and grinded them using mortar and pestle. Then we took the representative sample from each of the ground leaf sample, extracted that in 80% of the methanol in a rotary shaker using overnight. Then it was diluted and the observed range was determined using the spectrophotometer, and gallic acid standard curve was used in this purpose. For the sugar analysis, a representative sample of the grinded sample was taken, and it was extracted in 80% of the methanol in a hot water bath with 860 degrees centigrade. Then it was processed through number of centrifugation and filtration steps, and it was at last analyzed by HPLC MS. into CSF. For this purpose, glucose, fructose, and supersystem curve was used. To analyze our data, we used SPSS and we performed one-way ANOVA. Because our data made the assumption for ANOVA, our data were normally distributed and had homogeneous variance. Results for chlorophyll content showed that the older leaves have higher chlorophyll content compared to the younger ones. And there was a statistically significant difference between the chlorophyll. For the total phenol content, results showed that there was no significant difference between the age group 1, which is the older one, and 2, but the age group 3, which is the younger leaves, they have a statistically higher phenol content compared to the 1 and 2, i.e. the older leaves. For the sugars, results show that there is no significant difference in the glucose and fructose content between the age groups. However, sucrose content increases with age. When we look for the symptoms in the glasshouse for any visual appearance of the symptoms, we till the fourth and fifth weeks of continuous exposure to the rust spores, we didn't find any like distinct symptoms. But during the current week, the oldest leaves from the age group 1, which are 4 weeks older at the time of rust inoculation, they are starting to show some rust-like symptoms, but we are not sure if they are rust or not. Usually the rust spores, if they got favorable environment, they take 2 to 3 weeks to show the symptoms. But in our case, it took very longer and it's still not clear. So what we think the reason behind it is the temperature and relative humidity condition in the glass house were not optimal. Research shows that optimal temperature for spore production is around 19 to 25 degrees centigrade. However, in our condition, the temperature during the time of inoculation was not that optimal. It never went above 18 degrees centigrade. So that might be the reason behind that. As we haven't seen, like our main trial didn't go as we planned, we performed an additional trial as a safe way as well. So in that process, we took the legacy variety again and performed that in a controlled humidity and temperature glass house. We took a spore solution having 4.2 into 10 to the power 4 spores per ml of concentration and applied to the plant with the help of high mist spray. We covered the plants for 48 hours with the bags and we recorded temperature and humidity. And we are still checking for any evidence in the plants. So in our study, results showed that there is increase in the chlorophyll content of the plant leaves in the older ones compared to the younger one. This finding is quite similar to other research as well, and it has been explained that with increase in age, the photosynthetic apparatus of the plant leaves become more developed. Therefore, there is more photosynthetic activity in the plants. So that may be the reason for increased chlorophyll content in the older leaf. But for the total phenol content, it was found higher in the young leaves, which is again similar to some of the findings from other research. And it has been explained that during expansion, if the leaves start accumulating solids, this interrupts the synthesis of phenol in leaves and cause the low phenol concentration due to the dilution effect, maybe because the water content is also decreasing in them. And sucrose was higher in the older leaves, maybe because young leaves act as zinc and they do not synthesize most of the foods. Since they are not that active photosynthetically, maybe the sucrose is higher in the old leaves only. But there is a role of both glucose and fructose in the cross pathogen reaction along with the sucrose as well. no clear symptoms of rust infection have been yet observed, but there are some initial indications on the older leaves suggesting that that they could be more susceptible compared to the younger ones. To conclude, chlorophyll content and biochemical properties of the leaves vary between the different age groups. Higher phenolic content in the younger leaves may be the reason that they are less susceptible to the rust pathogen. Susceptibility of the blueberry leaves to rust pathogen could vary in leaf age. The earliest stage of leaf development may have less risk of infection. So, if so, fungicide application could be most effective if applied after the leaves reach the age of week 4. I would like to thank all the supporting hands during my research.
Any questions in the room or online?
Yes, yes.
I was behind you. Kalpana, good job with your presentation, thank you. I know it's very frustrating when you try and do your work in plant pathology, it doesn't work out. I just wanted to know, is there any sort of, can you go to a field which has an incidence of rust and maybe do field observations about where they are occurring first naturally?
Oh,
I honestly missed that in the background, sorry.
oh no! Actually, we took the rust like samples from one of the farm from the northwest Tasmania. So we just bring them back and then use hand inoculation methods with brush and just we brush the rust spores from infected leaves and brush them back to the each and every single leaves, as well as we had some potted plants which were infected initially in the glass house. We put the plants.
Yep, thank you. But I mean, I just wanted to know, could you observe the occurrence of rust in the field naturally, so you can try and work out a bit more about what age the rust, the plant is more susceptible to?
What is the first thing? I just.
Oh, sorry. I just want to know whether you could go to a field, an orchard, which has natural rust.
Oh, yeah.
Yeah, sorry.
Yeah, that would.
And then try to work out whether that could give you some clues about which age is affected.
Yeah, but because of the time frame and everything, it's.
So there's no literature? I mean, so.
Well, the blueberry rust there are very limited.
Okay, that's good to know.
Explain.
Thank you.
I think there's one online.
If the leaf is still a sink for carbohydrate, then what do you think that means for a fungus that is trying to source sugars from the leaf? A biotroph? Question
I
mark.
just want to see that question because. Actually, I have explained that in so in my research, I haven't found if the younger leaves are still like infected that way or not. So as I have explained, that working as a sink and they are not showing any symptoms yet because the older one are only showing where there is higher photosynthetic activity and more the sucrose and other things. I'm not pretty sure like exactly what's going on with that. Yeah, maybe.
Can I just follow up? I think what that question's getting at is why do you think these things like sugars matter to a biotrophic pathogen like a rust?
Oh, because for the biotrophs, they need some food in the forms of sugar like glucose, fructose to survive. They can't stay alive in the necrotic tissues. They need light plants. So.
Hi, you've measured various effects. How do you go about measuring interactions of effects? And let me maybe explain my crazy thinking. I question right now, if we went for a walk, how many leaves are there that are plus or 4 weeks old or new? Because we know there haven't been many days above 19 degrees yet this year or this season. And that's one example where, there's the possible interaction.
I cannot understand the reason.
Yeah, that's all right. That's how I talk. We'll ask during coffee. It's fine.
Thank you very much. So the next series of presentations would be from the honors students, and these two
Interaction of effects.
considered for the Art Institute of Australia Awards after today. And the first presenter for that would be Rhiannon.
Okay, so I am Rhiannon Cox, and my study is on effective enrichments in dairy calves under the supervision of Dr. Megan Burney, Dr. Tina Taylor, and Dr. Sarah Underwala. Sorry, I'm a little bit sick, so I'll try my best to open my voice days. Okay, so what is enrichment? A lot of people might think they don't really have any experience with enrichment, but if you've ever bought a toy for your cat or thrown a ball for your dog, you have actually enriched their environment. So well done. So if domestic animals are commonly enriched, why not farm animals? And what would that look like? So here's a picture from actually my farm. This is one of my pigs who's just given birth. She, most female pigs, before they go into birth, they'll have this like urge to nest and try to gather things and build their nest so that they can give birth in. And hay is one common enrichment that allows them to fulfill that urge, which is quite easy to do and easy to produce. So that's something that you can provide. Now, specifically to calves, what have previous studies told us? So a lot of the previous studies have been conducted in Europe and America, where the systems are quite different to ours. So they'll have mostly indoors for a lot of the year just because of the weather. And the calves are generally housed individually due to disease or the management strategies that they have. These pens are usually, provide for the bare necessities, but are often barren otherwise. And the studies have shown that a barren environment causes a negative welfare. Social enrichment is beneficial to animals, which is great because Australia, although having that quite hectic seasonal calving where hundreds of cows can give birth within 3 to 4 months, a lot of farmers will then house their calves in medium to large pens, which is a good form of social enrichment. However, cross-suckling is a major issue in these grouped house calves, and that's the behaviour where the cows having the urge to suck on something, as babies tend to have, they'll redirect that behaviour to their pen mates and they'll suck on ears or tails, anywhere on the body they can kind of get at, which cause health issues like hematomas, and it can even cause umbilical hernias with the belly button, obviously from a newborn baby. We know that higher welfare is good for the physiological health. On the production side, a lot of the time this means meeting targets like weight, so great for farmers. And then another benefit is the social license. So a lot of consumers love to see happy, healthy cows, and if they're buying the milk, they love to see that the calves are doing well. So that's another benefit. However, not all enrichments are beneficial. Despite good intentions, a lot of, some can even harm the animals if they're not handled properly. So that leads to the question, what is enriching? So we have the idea that in order to be effectively enriching, we are targeting our enrichments in this study to a particular behavioural urge. So to do this and to find our results, we're asking, will the enrichment be used? Will the enrichment alleviate stress? Will the enrichment benefit physiological health? That's the weight side. And does it affect development? So this is our pen layouts. We organised it so that each pen had 3, so the same number, just so that was kept constant. And because one of our enrichments had to be hung, we decided we'd hang all the enrichments just so that location wouldn't be a factor. These are the enrichments like close. So we have a cheap ball enrichment, which can be easily implemented. The hay one is obviously targeted kind of that grazing behaviour, a little more expensive. And then our star treatment, it's like a teething star. So they are kind of hard silicone edges for the calf to try and suck on. And then this is what they look like in the pen. So this is our mixed enrichment. You can see that there's one of each type. And as best we could with live animals, we balanced for sex, breed, birth weight, and age. So our behavioural observations, we set up GoPros in the corner of the pens to look in, and then I watched the footage from an hour before to an hour afterwards. This tends to be like the peak activity time for calves. And we recorded those at the week 1 mark, the week 3 mark, and the week 6 mark. That was the same for weights as well. We measured at the same times. So our recorded observations were targeting, did they use it? So we looked at if they interacted with the objects. Does it promote the welfare? So that's the play behaviours. You can even see that with your domestic animals. A lot of times people will say, oh, he must be sick, he's not himself, he's not playing, right? So for calves, that's a very good indicator of welfare. And for alleviating stress, we looked at the incidence of negative behaviours. So that's the chewing on the pen and then cross-sucking on pen mates as well. And long-term stress can affect the growth. So that's why growth is under alleviated stress. We did a novel object test at the very end, so at the 6-week mark, and we gave them a total of 5 minutes inside the pen, which was isolated from the other calves with a lot of high black plastic, and they were given 45 seconds before we put the novel object, which is just a witch's hat, to kind of acclimatize to the pen, and then full 5 minutes to see how or if they interacted with the object. This affects development, as a previous study has shown that the higher they wait to touch it, the higher their cortisol levels are at that stress incidence. Some calves really loved it and they decided to have a play with and just headbutt the novel object. Some calves are very eager and couldn't even wait until we dropped the novel object. So now for the results, where the enrichment's used. This is proportionate of the time used to the time observed for interactions with the objects. You can see that they all have quite high initial interactions, but as time kind of goes on, only the mix enrichment and the hay enrichment have prolonged use, and we saw a significant value with the, significant statistical effect with the p-value there. Our next one is reduce stress. So this is a physiological health. Obviously, if you're stressed for a long period, you're not going to put on weight effectively. So this looks at the average daily growth. We found that there's no statistical significance between the treatments with that high p-value there. And we did check for sex and breed. They didn't affect the average daily growth. Here we have the environmental exploration. So that's often the pen niggling side of it. So you can see that the ball treatment is continuously higher than other treatments, but there's a variability as the novelty wears off, but we did find an effect of treatment on that. This is cross-suckling. You can see that all treatments have a sharp increase in week 3, and that's likely due to the change in their food ration. They went from twice-a-day feeds to once-a-day feeding of double the amount, just because they were older, so starting that process, which is just due to the farm kind of practices. You can see that the stars didn't increase as high as the rest, but they all kind of did have a nice decline there as the calves got used to it, except for the ball treatment. But our significant value for that was in the interaction between the week and the treatment. So positive emotions, this is the play behaviour. We didn't find any significant differences between the treatments, unfortunately. So nothing there. But novel object testing. So we have no significant p-value yet. We do have another replicate currently being tested. So when that comes in, it might change as our pairwise analysis of the hay ball treatment did find a tendency for it to be a lower latency. But again, waiting for that replicate to come in. So just asking, is it effective? This is a table adapted from a 2023 review by Taylor and their colleagues, where we can do a nice cross and check. So we found that the ball did initially get that use of that positive hedonistic state. So they liked it initially, but no sustained use and no real effect otherwise. That's a pseudo enrichment. The hay enrichment, we saw that it had that positive hedonistic and sustained use. And again, waiting for that third replicate to really tell us positive mood or stress resilience, we think it might allow them to have that lowered stress reaction. So for now, enrichment for positive welfare balance. The stars, similarly to the ball treatment, wasn't enough by itself. So positive hedonistic state, no sustained use, another pseudo enrichment. The mixed treatment did quite well. We saw that it lowered the frequency of negative behaviors like chewing on the pen and allowed them to have a positive hedonistic state, and also had that sustained use, which was good. So that would be classified as enrichment for meeting basic needs. So the key takeaways here are that effective enrichments benefit calf health and welfare. And although our Australian standards, the social element of that is a good enrichment, there is more that can be done. And so I'd recommend producers try and investigate what enrichments they can add. The findings from this study recommend a nutritional type of enrichment like the hay balls, that was quite good, but also a combination to provide a more complex environment for their calves. But more research is needed to determine the long-term developmental benefits of enrichments, especially during more stressful times like weaning for calves, which occurs at the 8 to 12 week mark generally, but we did have to stop our study at the 6 week mark just due to time constraints. So thank you, and acknowledgements to all the staff at the Tea Dairy Research Facility for their help.
Questions for Rhiannon in the room?
I'm just wondering whether it was considered appropriate or not to have a treatment with no enrichment at all.
Yeah, that was something we did consider at the very start. We had a limited kind of space-wise. Obviously, you only have so many calves in each pen. But because of previous studies where they've shown that having no enrichments is not effective at all, and like quite a few studies showed that, we decided that it should be considered a bare minimum to have a type of enrichment in the pen. So that's why we didn't have a barren control. And yeah, but that would be something if we had more space, we would have put it in just to show.
Rob? Do cattle see blue? I'm just curious because you've got a lot of blue toys.
Yeah, no, that is a good question. We did have a slight difference with the hay.
Red hay
enrichments
ball?
as well.
Yeah, I saw that one.
Yeah, we had one that was a red and pink colour just because that's what the manufacturer had and they sent us. So I did look into colour studies. They are colour blind, technically the same way that dogs are. Blue kind of does stand out a bit more in the way that dark grey kind of stands out, and then red would be like a lighter grey. But in the differences that we kind of see and they would have noticed between like their novel object, there's not that great a difference. It's just like a slight, oh, that's grey and that's less grey. So yeah, not a huge effect on the study.
Good answer.
Yeah, great presentation. Thanks, Rhiannon. Just wanted to, you focused on dairy calves. I wonder if the behaviour or would be very similar or different when you chose, for example, a different type of calf, like beef or something?
Yeah, well, we had some that were crosses. So these ones, for example, here, that's she's an Angus cross.
Okay, so I thought it was dairy. Right?
Yeah, no, their mums were dairy. So I say dairy cows just because that's the environment they're being raised in. The main difference with beef calves is that they are raised with their mothers on an paddock, extensive paddock. So a lot of the time, those calves are just kind of left on a more natural, I suppose you would say, element. So there's less likelihood of cross-suckling. You almost never see beef calves cross-suckling because they have that free access to their mother, whereas dairy cows have that kind of limited feeding time. You get the morning feed and the afternoon feed if you're young, and then our calves changed at the 3 week mark. They got this full 6 litres in the morning and then no milk for the rest of the day, but they had hay and grain and water that they could go to. So that's why there's like the kind of difference in why you might need enrichments in dairy system.
Yeah.
How did you come to select the types of enrichment you used?
Yeah, so we had a thought about that too. We kind of wanted to target specific behavioural issues that might occur. So for cross-suckling, a huge one you see in dairy calves. So that's why we chose the teething style. We thought that might give them an outlet to kind of go to and express that behaviour so they can, redirect it away from their pen mates. The hay ball we chose to target to a grazing object. A lot of time in cows that can't freely graze, you'll see a lot of like tongue rolling. So in order to like eat, they'll roll their tongue around the grass and then rip it out. So we thought that the hay ball providing that kind of hole would allow them to stick their tongue in, roll it and pull the hay out. It also just gives them a new way to eat the same hay that is provided in a big lump. And the ball was something that had actually been used on the farm, just because some people were thinking, oh, well, it looks bare, let's give them something and see what they do with it. But it did encourage a bit of headbutting play. Sometimes you kind of see them do that, but that was one of the enrichments that we thought, well, it's very cheap, like those we bought at Kmart, like $4. So if it did work, that would be great, but it's probably something that can't be used just alone. So that's why we chose those specific enrichments.
All right.
Teaching me painting with the elephants.
Our next speaker is
I failed.
Hi everyone, my name's Ruby and today I'll be talking about the importance of microbial nitrogen fixation in dairy pastures. So nitrogen is an essential nutrient and it plays critical roles in nutrient cycling. It's required for a number of key processes including protein synthesis, chlorophyll production and energy transfer. And with limited nitrogen available, the crops can't grow and develop crops. So it's quite important in dairy pastures. So biological nitrogen fixation is conducted by microbes that convert atmospheric nitrogen into ammonia. This process is very important because plants cannot utilize nitrogen in its gaseous state. Nitrogen fixes are categorized into two main categories, which are free-living and symbiotic nitrogen fixes. Some examples of free-living include Azotobacter species and cyanobacteria. Symbiotic nitrogen fixes, most of them fall into the category called rhizobia, and they form symbiotic relationships with legume species and fix nitrogen via specialized organs called nodules. The nitrogen fixation reaction is catalyzed by the enzyme nitrogenase. The enzyme is synthesized by a range of different proteins. However, the NIF-H gene encodes an important protein that makes up the enzyme. And the NIF-H gene is therefore very widely categorized and used in lots of nitrogen fixation gene studies. So nitrogen fixation is important in dairy pastures as the pastures have high nitrogen demand to grow sufficiently. To keep up with this, often synthetic nitrogen is added in the form of fertilizer, and the cattle also produce a large amount of nitrogenous waste. So these two factors can lead to negative environmental impacts, meaning there is interest in reducing synthetic nitrogen additions where possible, as well as optimizing the use of rhizobia. There is plenty of literature available looking at the symbiotic relationship between the legume and rhizobia. However, there's very little related specifically to dairy pastures. So that makes my research important and relevant as well. So I had two main research aims. The first was to assess the diversity of nitrogen fixing microbes in dairy pastures, and the second to develop methods to quantify the potential of nitrogen fixation in soil microbial DNA using digital PCR. So to achieve these aims, I used two complementary approaches. So I got samples from the Dairy High 2 research farm in Elliott from farmlets 1 and 4. So farmlet 1 was a conventional pasture type
that had also had synthetic fertilizer additions, and farmlet 4 had no synthetic nitrogen inputs. So to assess the diversity of the nitrogen fixing microbes, I use culture-based techniques. I won't talk about this anymore today though. I'll focus on my second aim of developing digital PCR methods. So to do this, I first screened the soil samples from both farmlets for the presence of the NIF-H gene using conventional PCR. And then from those results, I began developing methods to quantify the NIF-H gene in the soil. So what is digital PCR? Nanoplate digital PCR is a molecular-based approach that quantifies nucleic acids. It uses Poisson statistics to calculate precise and absolute quantification of the gene copies in the target DNA. It's a similar setup to conventional PCR in that you make up the individual reaction mixes, and then those are pipetted into the nanoplate, and then inside the digital PCR machine, the samples are randomly distributed between thousands of different partitions. The sample is then run through a thermocycling process and then imaged using fluorescence, which shows if the target gene has been amplified or not. The digital PCR provides more precise results than alternative technologies, and you don't really need any expert training to be able to use it. And due to the statistical approach, there's no need for a standard curve, which similar technologies also need. It's worth mentioning there's very little digital PCR in the literature relating to soil DNA. So when I was developing my methods, I didn't have a lot of guidance. So for my first round, I ran 7 samples plus a negative template control just to check for contamination. I used the standard cycling conditions recommended by the manufacturer, and also an annealing temperature of 59 degrees, which was based off conventional PCR using the same primers that I'd done previously. You can see on the scatter plots there that the blue dots represent the positive partitions, and the grey dots are the negative partitions, and the fluorescence scale is on the y-axis there. So what you're looking for is a noticeable split between the positive and negative partitions, which you can't actually see in any of those ones there. So there's lots of noise in these results, what's often referred to as rain, which basically just means that there's suboptimal amplification of the target gene occurring. So this highlighted to me that the PCR conditions were not optimized yet. So to try and solve this, I lowered the annealing temperature to increase the PCR reaction efficiency. And I also included a positive control that was just NIF-H DNA, so it didn't have any soil. So these are the results with the lowered annealing temperature. You can see that the positive control up there has that nice split between the positive and negative partitions, whereas the samples don't. And you can see that the lower annealing temperature as well has caused a higher amplification of the target gene. But yeah, there's still lots of noise and lots of that rain. So the result of the positive control showed me that there's inhibitors in the soil samples that are affecting. So for the next round, I added a spiked positive control, as well as tried diluting the template DNA to try and dilute out the inhibitors, and used an extra DNA cleanup step as well. So you can see here that the spiked positive control indicates that there are PCR inhibitors if you compare the spiked positive control with the normal positive control. And then the dilution series, the most dilute sample, shows potentially that it's diluted out some of the inhibitors, but there's still a little bit of rain. There's not a nice split between the positive and negatives, and the ethanol precipitation didn't do much at all. So next I tried further optimizing the PCR conditions. So I lengthened the extension time and also added a restriction enzyme to some of the samples. So this is with the lengthened extension time, which didn't really affect the results too much. They're adding the restriction enzyme. You can see on the 2 scatter plots at the end there. You can see that nice split between the positive and negative partitions. However, there's only about 6 positive results. You need at least 20 is recommended. So potentially, if the template DNA was less dilute, we'd get higher amount of amplification. So that just shows a summary of my method development. The main issue was that I faced was the inhibitors in the soil samples. So whether there are alternative cleanup steps that I wasn't able to try, that would be beneficial. Potentially a C-tab approach, which is often used for plants, extracting DNA out of plant material, would be a different way to go. It has been used a little bit in soil as well. And I think restriction enzymes would definitely be worth trialing further, as those results were looking quite promising. There's plenty of different ones out there to try. Also, the NIF-H gene, there are many different primer pairs cited in the literature, and they all have varying success depending on what you read. So I think it would also be worth trialing a different primer pairs just to see if that would optimize the conditions further. And potentially, because the inhibitors were causing a lot of issues, maybe just using 16S primers to start with will help get a handle on the inhibitors before then trying the functional, targeting the functional NIF-H gene. So I found that nitrogen fixing microbes were present in both the conventional and no input farmlets. I isolated a broad range of nitrogen fixers. Therefore, I had to use the molecular techniques to target the functional NIF-H gene to try and quantify nitrogen fixation. And digital PCR, I think, does have the capability to quantify the potential of nitrogen fixation in dairy pastures. However, there is a lot more further research required to optimize these methods for use in soil DNA samples. And I'd just like to thank everyone that has helped me with my research this year, and thank you all for listening.
Right, some questions for me. Rob, we'll start with Rob and then.
This could be really embarrassing because I see my name on the slide there, and I was going to start by saying I know nothing about this. I'm curious about your scatter plots because I look at y-axis and its fluorescence intensity. So if you could help me understand a little bit, because I know nothing, you talk about inhibition, but you've got heaps of stars on the plot. And so it looks like you're getting a lot of hits. Is it like fluorescence quenching because you've got, your solution is too concentrated? What do you think is going on? I just try to help me understand a little bit if you can.
Yeah, so the fluorescence are good. Like you can see on the positive control there, you do want the fluorescence sort of up at that sort of 150 mark there is what you're kind of aiming for. But yeah, you can see on the ones that have heaps of those partitions that have amplified, probably in that case, there's just too much template DNA and the inhibitors are being, causing that kind of.
Or is this some kind of native fluorescence from, because you've got a very concentrated solution?
Yeah, potentially. I was mainly focused on trying to get rid of the inhibitors, so I'm not actually sure, what.
Certainly looks like you're heading in the right direction when you start to dilute. It's really,
Yeah. So I think diluting them out, whatever it is, there could be things in the soil that are, fluorescing.
nice.
One question.
Thanks for the presentation. It's really nice. And maybe related to the question, because you're kind of targeting all the plausible things about hitting the inhibitors. Do you have an idea what the inhibitors might be? Because you can either dilute or you can just hammer them.
Yeah.
Get rid of them with some sort of treatment. What are your thoughts on that?
We had sort of, we're trying to get rid of them because we actually originally weren't really expecting them to be such an issue, or for there to really be for them to be there. So we didn't really know what they were. So I was mostly focusing on trying to dilute them out or try other things that would get rid of them, but not exactly sure what they are.
Thanks.
Thanks very much, Ruby. That's a great presentation. This is actually not specific to this particular presentation, but I just wanted to know out of curiosity, you compared the two types of systems from your analysis of all you've done. What is your sort of gut feeling about nitrogen fixes between the two systems?
So the samples that I have been testing are just baseline samples, so it's right at sort of the beginning of the farmlets have been set up, and they're actually not. There's not really any difference at the minute between the conventional and the no input farmlets. So it's hard to sort of say. I think it'll be interesting to see as it goes on, seeing how it plays out, because potentially at the minute there's sort of things in the soil that were already there before, like there's base level nitrogen and things like that. But I'm interested to see how the no input goes and see if
I could butt in and just say, keep in touch with Ashraf, because he's going to be working on the project for a lot longer.
Sorry, just a quick question about, do you know the history of the paddocks? Were they like similar before the trial began? Or?
I actually am not 100% sure. Sorry.
That's okay. Obviously, you're looking at function, and so it's important to look at what's actually happening in the soil. But would there be any relevance to be working with root samples instead of soil samples? Because you might find less inhibition.
Not specifically for this, I just was using the soil samples. But for the culture-based methods, I used both soil and I took the root nodules out and looked at the bacteria that were in the root nodules as well. So that could definitely be something to try here. I just had issues actually isolating nitrogen fixes from the root nodules themselves. But if you could successfully do that, it would definitely be worth trying. Yes.
No more questions. Thank you very much, Ruben. Thanks everyone for staying by with us up until this point. We are going to take a break for morning tea, and we will be back to continue with 4 other presentations at 11:15. So there could be some coffee and maybe for something for morning tea out there. So you go there and have some, interact with those that are presented, and then I'll see you out. Thank you.
Are we okay? All right, everyone, welcome back. Thank you for joining us again. I'd like to introduce you all to Mohit. Mohit has disappeared. He's coming back. We have technical issues and I need to entertain you for a few seconds, so.
Sing a song.
Tell a joke.
Mammy, oh, mammy. Al Jolson. That was the first song to be recorded, film and sound, in the history of humanity. Al Jolson's Mammy. I don't know if he had a hat in his hand at the time. Are we okay now? Yep. Okay, let's start again. I'd like to introduce you to Mohit. He's one of our master students, and he's going to talk to us about Beetle ID in strawberry. There you go.
Hello everyone. I hope you are fresh now because of the short breaks. And let's take a dive into small insect world. My name is Mohit, and today I'm talking about the novel pest of Tasmania, which is we call it the strawberry beetle. So it is really important for the Tasmania. We have many of berry growers. We know that Costa berries, Hillwood berries, Wellington berries, and Costa berry as well. So Tasmanian berry production around $99 million in 2018, and which is accounted 15.9% of national berry production values. So today I'm talking about this beetle. In recent days, we spotted these beetles in northern Tasmania, especially from East Devonport. So emergence of new pests we call as a Dicus arta, the strawberry beetle. Sometimes it's referred as a bronze leaf beetle. Member of Chrysomelidae family. There is found in some Australia, Caribbean, and other parts of Asia, just like Japan as well. In Australia, it's reported on 1988 in legumes by scientists Reid, and recently we spotted on East Devonport. So why we concerned about this beetle? We don't know about anything in beetle. Last research has been done on 1949 in the USA. So we don't know about the potential crop damage and economic import in strawberry. We don't know about a host of the pest and habitat as well surrounding the Tasmania and surrounding Australia as well. We don't know about anything, especially on the biology, because it's really important for any insect to complete their life cycle. And if we don't know about anything, especially we don't know about management strategy as well. And sometimes we don't know either it's a threat for our biosecurity in Tasmania as well, or either we concern as an invasive potential pest or not. Who knows? We know. So in this research, I'm trying to solve these three questions, especially what is the complete life cycle strawberry, especially in Tasmania, because we know that it's cold environment. What is the nature and extent of the damage caused by this beetle, especially in strawberry crop, and if we found any either host crop in Tasmania as well. What is the most effective and sustainable control measure available for the managing this beetle? So for my first objective, we collect the beetles from Costa Berry at Devonport, and we just collect the beetles sufficient amount and transfer into the entomology lab at the Sandy Bay. Then we set up the rearing box, which is rearing box is over there. We just close the box with a mesh bag, and we don't know about anything, so we just prepare some layer of the soil. We just it's just like hypothesis that either eggs lays on the upper surface of the crops or plant parts or in the soil as well. So we did that, and every box has one female. And for food source, every day we just provide a fresh strawberry leaves and flower as well. For the life cycle, egg larva, pupa were separated into different boxes and vials, and then we just noted the date of each stage. For my second objective, for a potential damage, we just collect the beetle same as what we require for that one. Then we select the flowers of 50 flowers we require for that. 25 flowers covered with the mesh bag, and one beetle per one flower per plant. We just bagging over the flowers with the insect, and on this side we just cover with the bag, but we are not introducing any kinds of beetles. Then after harvesting observation, we will do two kinds of observation: weight and quality of grading. So on this objective we are doing right now, and we haven't get any kinds of strawberry right now. We will do that in short days. For data analysis, we conducted TTS and compare between fruit qualities and the weight, and with the control group as well. For our third objectives, we go with Australian pesticide and veterinary medicine authorities. We find some literature reviews. Especially, we are main focus on biological controls on non-chemical exclusive control if it is available. So now we talk about the result of biology of Dicus artaris. You can see the eggs lays on the undersurface of the leaves. Then it's transferred into the soil. So you can see the cover over there, which protect from enemies as well. Then a further next stage, it's in the cover. They set up for the pupation stage, and then after they become adults. Fourth, we can talk about the average of life cycle. It's around 19.4 days, with a maximum of 100 days or minimum of 10 days. Now we talk about the seasonal availability. So we just mapped down East Devonport berry map, and we can see that a red circle, which is known for is a higher population of the insect. It's around tunnel number 1, 2, 6, 6, 5, and 8, 9. And for the seasonal abundance, we can see that in 2022, 2023, and 2024. In the December, we have seen some spike over here. That means it's influenced with the temperature and humidity as well, because at that time we know that in Tasmania we have higher temperatures as compared to the other month. So beetle population increase in November, December, and January. However, they are active in all around the year. If we talk about the potential damage, so we can see that beetles is over there. So adults damage to leaves, flowers, and initially they feeds on leaves. Then after flowering comes, they transfer into the flower base near to the style and stigma. And sometimes they do a shoot hole appearance on the leaves and some bird petals as well. They feed on base of the flower near to the style, consuming tender parts. They also sometimes seen on the fruit as well, and which is make unconsumable for the consumer as well. Grubs also cause damage feeding on leaves, sometimes on the flower as well. If we talk about the result, we found 5 different types of chemical, but it is not on strawberry. We found on all around the robust species. First, we talk about the malathion, so which come from organophosphate class, which has knockdown effect and relatively low toxicity and for the beneficial insect. But in the contrast, bifenthrin, which is class for pyrethronoid, has a long-lasting effect control, but it should not be used because of its impact on beneficial controls. And Costa berry, they release beneficial control insect in their farm. And most of the berry growers, they are doing right now. For chlorpyrifos, which is also again stood on the organophosphate class, historically it's effective, but has same as environmental impact on our health risk as well. Indoxacarb is very effective as well, safer for beneficial insect compared to other 3 options over here. Pyrethrum, which is very good because it's derived from chrysanthemum flowers, and it is environmental friendly and minimal impact on our beneficial insects. So malathion and pyrethrin are recommended to our growers. They use against this pest. For our summary, the beetle completes life cycle in average of 19.4, 5th and 5.58 days. From November to December, they are very highly active in growing their populations, but throughout the year, they are active as well. Both adults and grub, they damage the flower fruits and which makes unconsumable. Malathion pyrethrin are recommended for controlling this insect for growers as well. And let's see what our second objective results comes, because we are doing about the potential damage as well. But it's open our future doors for the research. Population dynamics of the beetles. We also do economic threshold level as well as economic injury level. So it's really helpful for growers to set up their benefit and cost ratio to their farms. And also we require for bioefficacy of newly insecticide on, especially on this beetle and especially on this crop as well. Moreover, we also look into the biological control. Either we have nematopathogenic fungi or parasiticide or parasite as well, because we don't know anything about this beetle in the world as well. PR team, especially John and Lynn and Ravinka from Costa Berry and another very Costa Berry farm members as well. Thank you.
Questions, please. Shane?
So you said you were going to look particularly at biological control and exclusion
Yes.
methods. So you sort of mentioned
It is
there that,
available.
yeah, you mentioned at the end that biological control is something to keep looking at. Did you find anything about other non-chemical?
We hardly found only one research paper regarding to this.
Yeah, okay.
We can't find anything around this area. So
Back
yeah.
to chemicals for the
And
moment.
yeah, right now we have only a chemical solution right now. But in the futures that I mentioned that it just opened the door for biological controls and bioefficacy, either rational for the newly insecticide as well.
Okay, thank you for presentation. Good job. My question is, just to look at at a different level, it seems to be like a pest that's emerging.
Yeah, right now it's emerging.
What do you think are the main factors that are causing it to become a pest? Like previously, maybe it wasn't not a pest. So what sort of things do you consider contributing to become a pest?
Especially on my second objectives that I mainly focus on the production, because if production impacts the grower, for the growers, definitely it will impact on economical way as well. If it is not too much damage, then we cannot consider it as a pest. But right now, the Costa Berries, they suggest that, okay, we just reduce our grading systems, and it's also ineffective, any kinds of chemical, but this is the problem right now in the farm, so we have to look into that as well.
The question is, what has changed in the last few months or years that has resulted in this insect becoming a pest when it wasn't previously? What are the causes? Is it change of climate? Is it?
Yes, as I mentioned.
Practices?
Yes, thank you. As I mentioned on previously, it's depends on the weather, because we know that in January, December, and November, we know that temperature goes higher as compared to our previous month, especially in Tasmania.
So you think the change in maybe an increased temperatures?
Changing in the weather, it's directly impact on pest population.
Is this particular pest specific to the strawberries that are grown on the tabletops, or are they not? Are they present also in the ones that are grown in the ground?
I haven't seen any in the ground on any host plants, because we did a survey with other berry farmers.
So you think it's more of the tabletop strawberry grown plants?
Yes.
I think they're all tabletop grown now, pretty much.
In Sydney.
Yeah, in Tasmania,
There's still some grown.
Yeah. All the Costa grows are grown on
Interesting,
table.
Alida.
Yeah, sure.
Good question. That's very interesting. Any more questions? Anyone?
Maybe just briefly. You mentioned, I think I heard correctly, you said that this was researched once upon a time, but nothing since about the late 1940s. And so, there's two ends of the continuum that might have led to that. Well, it's out of control. There's nothing we can do about it, no matter what we do, or it's not such a big problem, or somewhere in between. Where is your, and I know this is research in progress, but from a gut feeling, where do you think we are on that continuum?
So from my gut feeling, we can't find any kind of result, right, because it's going. So from my perspective, it's a hypothesis from my side. I feel that it's not effective as much as economical way, but the population growing in some months, it's really damage on the flowers and the fruit as well. So yeah, at that time we consider it as a pest as well. But I've been to previous data as well. Population present around the world, but they're not considered as a pest. So they count as a beetle for whatever survey they are doing, but not considered as a pest. But right now we have seen many damage on the flowers and fruits, and that's why we are conducting the research.
Do you reckon it's going to accelerate in the
Yeah.
future? Okay. All right, any more questions? Anybody else? Well, you've either scared them or impressed them very much. I think you've impressed them very much. So everybody around the room. May I invite, oh, I've got to get the presentation off us. Please relax. I'll get the next presentation up.
Yeah.
Ram, may I ask, invite Ram up here, please? Thank you. I'd like to introduce you all to Ram, another one of our master students. His title, the project is Potential for Growing Red Rooted Sage as a Traditional Chinese Herb in Tasmania. Yeah, try it. Can you try it? Just good on you. All right, thank you. Go ahead.
Good morning, everyone. I'm Ram. I'm master's student and doing this project is a part of Masters of Agricultural Science. And the project title is whether we can grow the red root sage Salvia miltiorrhiza in Tasmania or not. And I'm doing this project with the help of my supervisor, Dr. Elita. The traditional Chinese herb, Salvia miltiorrhiza, is used for treatment of different diseases. It is commonly called as a red root sage, and its dried root is called as dansen in China. The Chinese name is dansen, and the wild population of this Salvia miltiorrhiza is mostly grown in the broadleaf deciduous forest in central and eastern China. The average temperature and pulse temperature range between the -14 and 31 degrees Celsius in the native growing region, that is Shaanxi region in China. And red root sage should be the well suited to growing under the Tasmanian climate and soil types. And here is a 2-year-old Salvia miltiorrhiza that was grown in Devonport and in the Forsyth Research Station. And there is climatic comparison between the Devonport and Shaanxi. The Devonport, where we grow the 2-year-old Salvia miltiorrhiza that was harvested by TCM Research Hub. And the climatic comparison, the rainfall pattern shows that there is similar kind of rainfall, and the highest rainfall occurred in July in the both region. The rain in Devonport, the rainfall was distributed throughout the year. However, in Shaanxi, there is no rainfall in the winter season. And talking about the monthly mean temperature, in Devonport, July is the coldest month. However, in Shaanxi, the July is the hottest month in Shaanxi. These are the 4 important bioactive compounds in red root sage. They are salvinilic acid B, tannisonin first, tannisonin secondary, and cryptotannisonin. According to the Chinese pharmacopoeia, the concentration of salvinilic acid should not be less than 3%, and the total concentration of tannisonin first, tannisonin second, and cryptotannisonin should not be less than 0.25%. Here we start talking about post-harvest treatment. Generally, this herb is used in a grounded form or dried form, and there are quite limited research on the post-harvest of this medicinal plant. And traditional post-harvest typically involve the oven drying and steaming that has to reduce the enzyme activity and which prevent the loss of active biocomponents and degradation of quality. The main post-harvest treatment for Salvia miltiorrhiza is drying at 50 to the 120 degrees Celsius temperature and steaming for 5 to 10 minutes, but steaming is less common. And appropriate steaming and drying treatment have been shown to help increase the concentration of major bioactive compound. And project aims, the first aim of our project is to find out whether we can grow the red root cells in Tasmania or not. And second aim is to study the effect of different post-harvest treatment like steaming and drying on the root chemistry. And hypothesis, steaming red root cells will improve the composition of major chemical compounds, and drying treatment, freeze dried and oven drying at 35, 60, 105 degrees Celsius may influence the chemistry of red root cells. Materials and method. The 2 years old harvested red rooted cells was provided by TCM Research Hub, and it was, after harvesting, it was kept in the freezer till the start of the lab work. After starting the lab work, I sliced the roots of Salvia miltiorrhiza using the glytin, and about 1 kg of slice sample. I have just around 1 kg of slice sample, and I divided the sample into 2 groups, just in the half. And 1 group of the sample was steamed in water bath for 10 minutes. However, other group of sample was not steamed. And after steaming, all the samples undergoes the different drying treatment like freeze drying and oven drying at 3 different temperature, and other non-steam one also goes the same treatment. And after drying the sample, then it is crushed in the ball mill and changed into fine powder. And after crushing the sample, it was extracted using the 70% methanol. It took 2 days to prepare the extraction solution for SPLC analysis. And after that, it was analyzed by SPLC, and Jacob helped me for the SPLC analysis. He's a PhD student working in a PhD in Research Hub, TCM Research Hub, and he helped me through the developing the SPLC methods. And here is our result. 2-OA ANOVA was used to analyze the result and to see the effect of steaming and drying. And 2 standard solutions, commercial standard, were used to identify the presence of tannin in second A and salvinic acid B. In the graph, we can see in the y-axis, we can see the concentration of bioactive compound. In the x-axis, we can see the different treatment. In the first graph, in the y-axis, there is salvinic acid B, and steaming and drying was found significant. In all the steamed sample, the concentration of salvinic acid was found higher than the minimal standard level, that is 3%. In all the steamed sample, it was found higher. However, in the non-steamed sample, only in the case of freeze dryer, the concentration of salvinic acid was found higher than the minimal required level. However, other treatment did not show the effective amount of or good amount of salvinic acid B. In the second graph, drying was found significant, and the y-axis of the concentration of tannin in second A, and here oven drying seemed to be reduced the concentration of tannin in second A. However, other treatments show the nearly equal amount of tannin in second A. In the third graph, there is concentration of tannisonin first in y-axis and in the x-axis the different treatment. And drying was found significant here, and the concentration of tannisonin first was dramatically reduced in over 105 degrees Celsius. However, the concentration was higher in other treatment. And in the last graph, the y-axis shows the concentration of total tannisonin. This is most important things in the case of this medicinal plant because it is considered as a total. We have to consider the total concentration, and drying was found significant. And at all drying treatment, the concentration of tannisonin, total tannisonin, was found lower than the minimum standard level. And also at oven drying temperature of 105 degree, the total tannisonin was found in the minimal amount. And talking about the cryptotannisonin, it was not detected in all the drying treatment. In conclusion, concentration of salvinic acid B was found higher than the minimum standard for all steaming treatment. The concentration of total tannisonin first, second, and cryptotannisonin did not meet the minimal standard for all drying treatment. We are going to examine the concentration of tannisonin by using the 100% methanol, because we have used 70% methanol in this extraction process. And steaming significantly increased the concentration of salvinic acid B in comparison to the non-steam sample at all drying treatment. At last, all these findings suggest that we can grow red root sedge in Tasmania. However, we have to follow them with effective post-harvest treatment to obtain the critical standard level in chemical quality. At last, I'd like to thank Dr. Eristalis for helping me throughout this project, and Jacob for he helped me develop the SPLC method and for analysis, and Caroline Clay for providing the access for the lab and different lab equipment, and TCM Research Hub for providing me the research material, and AEMG. Thank you.
Questions, please. Yes.
I didn't see your first slide, Ram. Is steaming usually part of the production method in China? Is steaming usually done in China as part of the production method? It's a normal part of the method?
No.
No, so you just added that because you...
Yeah.
So what was the reason for adding the steaming? Why did you think steaming would be a good thing to do?
Yeah, in few paper I have seen that steaming boost up the concentration of different important bioactive compounds. Just like salvinic acid.
Yeah.
It is not common. It is not commonly used, but however, in some paper have done that. So I like to test that one. So I did the steaming here.
Yeah. Do you think that might affect its marketability to the traditional Chinese market?
Yeah, sure. Because it has to boost up the concentration of different compound and its effectiveness. So it can affect the market.
So the question is, do you think it will affect the marketability of the product to the Chinese public? So John's question is, will it enhance the sale of this product in the Chinese market, steaming?
Yeah.
Will it enhance the sale and promotion of this product?
It is less common, but it may.
It may. Well, I think it would, because there'd be more of the compound of interest.
Yeah, I mean, I don't know much about some people in the room, but I guess sometimes there's a tradition in how these things are prepared, and they may not want to move away from that traditional method.
Absolutely. Sorry, ladies first.
Yeah.
Go
on. Just wondered, you showed the effect of steaming on that first compound, but then you didn't show it for the other compounds. You just showed the actual drying treatment is.
So you showed the steaming, sorry, you showed the steaming for the first chemical, but you didn't show the effect of steaming for the other chemical. Why is that?
Because steaming was significant difference.
Because it wasn't what?
Answer.
It didn't, because it did not any significant difference between this.
Oh, okay. Okay, sure.
So, yes.
I just got an online question.
Oh, sorry, but Arpes first. Yeah, sorry, Arpes.
Yeah, so I think you mentioned that you were going to look at whether we can grow this in Tasmania in comparison to China.
Yeah.
So did you do anything like that? Because I didn't see you mention anything about it in your result.
Oh, that was 2 years old plant. Yeah, it's talking about.
No,
did
no,
you-
you, can you repeat the question?
So
Arpes?
in your objective, you wanted to know if we can grow this plant in Tasmania.
Yeah, we can grow.
So did you work on that objective in this study? Because you didn't present anything on that. That's one.
Just the material was provided by TCM Research Hub, and just I analyzed the sample.
So they
That was
didn't.
2 years old
So
sample.
the question is, Ram, basically by by looking at the chemistry of the material, we can say that yes, we can grow it in Tasmania or not. Because if we don't meet a certain level of chemical bioactive, then it's not suitable to grow in Tasmania.
So it's not a factor of whether the weather or
Yes, it's
weather
both.
condition
It's
is
both. Yeah.
because I was thinking that maybe there was a study or a trial that compared Tasmanian weather to Chinese weather in terms of growing the product. But yeah.
Online question, Alison?
So what do you see as the effects of steaming? Is it to prevent metabolism or does it foster conversion of one metabolite to another? Would you like to read the question?
What question here? So what do you see the effect of steaming? It prevent the enzyme activity. It prevent the enzyme activity and reduce the just change of the one chemical to other chemical form. And second question, say, you see the effect of crop loss concentration. Yeah, it really affect the concentration of chemical because the climate and temperature is the main thing that decide the plant growth and development. So here is the result. We found less tannisonin. That may be the result of the climatic condition. Yeah.
May I put in as any more questions? Rob.
So compound seems to be thermally labile. You see 105 degrees, your recovery is like really dropping off. So steaming seems to be really counterintuitive, which is just a comment. My question is, freeze drying seems to be effective, but it's very expensive. And so what would your recommendation be? Because if we can make something, but it costs 25 times more to produce, are we going to be able to sell it? What's your recommendation of these drying approaches?
Overall, my recommendation for the drying process is just steaming the sample and then drying it, oven drying around 60 degrees Celsius, maybe quite effective. Because 105 shows the reduced level of concentration of chemical. Steaming and 60 degrees Celsius, maybe.
Well, we're guessing it's about enzymatic rates, and the steaming cuts off those enzyme activity much faster than a slow ramping up in an oven.
And so drying, is that in the presence of oxygen or?
It's
when you
drying
dry,
in the present, is there oxygen when you dry it? Is there oxygen around?
or in inert atmosphere?
No, it's oven. There's oxygen, it's just a normal oven.
Oven temperature.
Okay. All right, any more questions? Oh, thank you very much for your time. Okay, I'd like you to introduce you all to Ms. Ramida, and she's going to talk to you about impact of cold storage and second grade fruit on the quality of freeze-dried berries. All yours.
Thank you, Adele. Good morning, everyone. And my name is Ramida, and today I would like to bring you through the impact of cold storage on the physical, chemical, and sensory properties of freeze-dried strawberries. Now, let's get started with the background. Every year, Australia has 7.6 million tons of food waste, and 70% of them are from edible part. Based on the study in 2017, tomatoes and other soft skin produce like strawberries were rejected by the retailer up to 88%. Why? Because it's outgraded the aesthetic standard. Additionally, according to the study in 2023, they had estimated the post-harvest loss of the strawberry up to 23%. And another reason why is because the strawberries are highly perishable and susceptible to mechanical injuries. And I will explain why this number is so important. In terms of the economy, strawberries has a production value at $429 million, with a household consumption up to 72% in last fiscal year. And strawberries, as you may know or not, they are rich in nutrients and antioxidant capacity, and they're also importantly tasty. With their natural characteristic, as I mentioned previously, they easily get dry roots, damage, and other aesthetic defects, which then they are degraded to second grade food according to the criteria that has been studied and summarized in 2017. And to reduce this waste, increase their value, and continue the pipeline in the market, the second grade strawberries are transformed into other kind of products like strawberry jam that you see in the market on the shelf. And freezing is used to retain its properties. But however, freezing just only reduce or slow down the biochemical properties by changing the free water in the system into ice particle. With the cell structure damage from the ice particles, the change in chemical compositions and sensory properties occurs, especially in the long-term storage. For example, antioxidant compounds like total phenolic content or the anthocyanins may increase or decrease over time, and we may get ethyl acetate, which is the pineapple-like aroma that may cause the off flavor in strawberries. And to maintain and retain these quality properties, freeze drying is used. And what is freeze drying? Freeze drying is a drying process that removing water from the product and is suitable for fruit and vegetable. And water is removed by changing from solid phase or the ice crystals into gas phase, which we know as the sublimations. With the freeze drying, the removal of water can condense and retain most of physical, chemical, and sensory properties. And for example, it may increase some of favorable compounds like furanol and sweet caramel-like aroma compounds in freeze-dried strawberry. And according to the background, we are still unsure how freezing techniques impact the physical, chemical, and sensory properties of freeze-dried strawberries that come from the second grade fruit. And also, what is the suitable storage temperatures and maximum storage durations that we can still deliver the high quality of freeze-dried strawberries to the market? And to answer all those questions, we investigate and evaluate the impact of freezing techniques, freezing storage temperatures and durations on the physical, chemical, and sensory properties of freeze-dried strawberries. Now, here come to the methodologies. In the methodologies, the red Rhapsody strawberries or red Rhapsody were used in this experiment, and they are sourced from a local supermarket because it's strawberry off season in Tasmania. And the strawberries were from Queensland, and they were harvested on 3rd of August, and our experiment was started on 13th of August. And it is 10 days away. According to the researchers, the optimum duration for the fresh storage is between 1 to 5 days. And the samples that I got have already got dry bruise, damage, and uneven ripen, and also other aesthetic defects like uneven shape and irregular shape upon receiving. And these fall into the criteria of the second grade fruits. 2 different freezing techniques were used: irregular freezers and air blast freezers. Quick freezing techniques using air force to identify the impact of initial size of ice crystal formations in the frozen strawberries. And the faster the freezing rate, the smaller ice crystal form. And as mentioned in the background, storage temperature and durations may impact to the quality attributes in freeze-dried strawberries. So 2 storage temperatures were studied with maximum storage duration at day 60 due to the time constraint. And the samples were tested on their physical, chemical, and sensory properties according to the table here. And here is the flow chart of how samples were prepared. And we use a laboratory freeze drying unit with a condition set at -76 degrees Celsius for 72 hours. Now let's move on to the result and discussion. We started with the physical chemical properties. There was a slight change in the pH, but insignificantly different. However, the freeze-dried strawberry at -80 degrees Celsius here show the lowest rehydration rate. Why? This may be caused by the storage temperature at -80 degrees Celsius and the air blast freezing techniques that inducing smaller ice crystal formations and resulting the less structural damage and smaller pore morphologies, which is require further study in terms of the individual morphologies and also textural analysis. Now come to the antioxidant part. Total phenolic content increased in day 15 and day 30 of the storage before significantly lost in day 45. And -80 degrees Celsius show the better retentions of phenolic content. The change in the compositions significantly correlated to freezing techniques and a combinations of temperature, storage temperatures and durations. The degradations is caused by the activations of oxidative enzyme during freezing and drying process. This is caused by the cell breakdown from ice particle that induce a higher extractions of the phenolic compounds. And the more availability of the compounds, the more degradation occur in the system. And this also causes a similar trend in the anthocyanin content in the next slide. Anthocyanin overall increased by 30% in day 15 and decreased in day 45, which -80 degrees Celsius also show a better retention in the anthocyanin. And in this case, the size of ice crystal formation regulate the change in the amount of anthocyanin during freezing and storage. The larger size cause more damage to cells, resulting in higher extractions. For example, in control freeze dry samples that we use the regular freezers. However, the longer storage may cause the degradative enzymatic reactions and increase the availability of the reactive anthocyanin compound in the system, resulting in the loss of anthocyanin in day 45. And the change in the compositions of total phenolic content and anthocyanin led to the significant change in the antioxidant capacity. The antioxidant capacity measuring with 2 different methods show a difference in sensitivities and the reactions of biochemical compound, which 15-day storage show the highest capacity of the ferric reductions, while there was an opposite trend in scavenging reactions. And one of the observations is that the initial irregular trend occur, which was possibly caused by the long freeze-dry processing in the control sample longer than 72 hours, that might cause a different pattern in chemical reactions. And based on the data, further study will be continued in the day 60 to confirm the antioxidant capacity trend. Now come to the interesting part, the sensory properties.
Clicking the wrong way.
Oh, thank you, sorry. Yes. Thank you. And then come to the sensory part, the color. There is a slight change in the color of the freeze-dried strawberries, overall slightly brighter and more vivid. This may be caused by the removal of water molecules that might intensify the color compared to the fresh strawberries. And at low temperatures, it prevents the sugar degradations, which is the cause of color darkening in the strawberries. And this is a photo of fresh strawberries compared to the freeze-dried strawberries over time. And the last part is the aroma compounds. Overall, 133 aroma compounds detected in freeze-dried samples, which each sample present same or different compositions than the compounds. Alkanes is the largest group in freeze-dried strawberry, giving faint, waxy odor. The key aroma compounds like ester, ketone, furanone, and lactones increased, giving out strong, fresh, fruity, sweet aroma. And the most interesting compound is the furanone. That has the low odor threshold, giving out sweet caramel-like aroma. And the large percent of furanone was observed in freeze-dried strawberries at minus 80 degrees Celsius, giving out the best odor here. And this might be caused by the change in biochemical reactions and moisture content during storage and freeze-dry. In the continuing work with the result from the study, the impact of 60-day storage will be further investigated next week. And due to the time constraint, future works and research are recommended here. So structural porosity, texture analysis, sensory analysis for consumer acceptabilities, microbiology and water activities, because the second grade strawberry have high risks of fungus and microbial growth, and also extending the study to actual farm gate with the different strawberry cultivars. So in a nutshell, freezing techniques, storage, temperatures, and durations have the significant impact on physical, chemical, and sensory properties of freeze-dried strawberries. Based on the current study, the optimum storage durations and temperature is between 15 to 30 days at minus 80 degrees Celsius with air blast freezing techniques. And last but not least, the strawberry cultivars' degree of ripeness, post-harvest management also influence the initial amount and compositions of the biochemical and aroma compound that may subsequently change during frozen storage and freeze drying. And before ending the presentations, I'd like to acknowledge Tia and Utah for the funding and opportunities, and my supervisors teams, especially Sai, Jay, Naomi, and Sam, and every doctor, professors, and friends for helping, guiding, and shaping up this project with me and my family for their support in my well-being. And here are my reference, and thank you.
Questions, please. Questions. Alida.
Alida, thank you for the presentation. You did a lot of different analysis for sure. Yeah. I'm really surprised that the antioxidant content reduced at minus 80, even after 45 days. What do you feel will happen a longer time storage? Strawberries or frozen strawberries are sold on the basis of the fact they're healthy, but even after 6-day storage, they're losing their antioxidant properties. I'm sure by the time a consumer gets the strawberries, it's going to be longer than 45 days. What are your thoughts? Are you still recommending strawberries?
Sorry, repeat the question again.
Sorry.
I couldn't capture.
The strawberries are sold because they're healthy, and when
Yes.
they're frozen, they're supposed to be still healthy.
Yes.
But your study shows that the antioxidant properties reduce.
Yes.
Even after 45 days. I feel that maybe the concentration will go down even further after longer storage.
Yeah.
Are you still recommending that we buy fresh frozen strawberries for their health benefits?
So first part, so as I explained, freezing the strawberries, it doesn't stop any biochemical reactions inside the strawberries, but it can slow down and to even like slower down those reactions. That's why we remove the water, free water molecules in the strawberries to retard all of the biochemical reactions that might potentially happen in that. And yes, and to answer your questions, do I still recommend? Yes, I still would recommend people to enjoy the strawberries because there's still a lot of things that going on there, like phytophenolic contents and also anthocyanins, which is really good in terms of the in some literature they show that they have the anti-cancer, anti-inflammable, and also
Right, thank you. That's a good answer.
So,
yes.
Do you think your results would have been improved if you'd have freeze dried them earlier? Because you said they were 15 days old when you started sampling. If you'd have done that at the source, do you think you could have prevented some of the losses that you had? Because your strawberries were, what did you say, 15 days old when you started sampling?
Yes. The answer is also yes and no. It's because the strawberry itself, the nutrient inside, it depends on how the farm grow the strawberries. It depends on the climate at that time, because in some literature review that the hotter the climate, sometimes they store more nutrient inside themselves. So yeah, we have to like check at the farm how they grow the strawberries, and then we can give the answer like, is it gonna be better or not? Yeah.
I think I'm quite confused. See, I'm not even sure that I'm confused or not, but I think I'm confused. When you talk about these storage times, is that time frozen or is that time freeze dried
It's
and stored?
Frozen.
Right. And so do you have comparison of frozen versus freeze dried, or? So everything is freeze dried after a certain time.
Yes.
What is stability like of freeze dried strawberries?
What is?
Yeah. So when you have a freeze dried strawberry, this one here,
Yeah.
how do all of those physical and chemical parameters track over time? Do they continue to reduce, or does freeze drying stabilize the material?
So to answer your questions, unfortunately, I didn't do the comparative between the frozen strawberries and after it's freeze dried. So I cannot give you that kind of exact answers, but based on the data that I get here, after subsequent storage time, there is something going on inside of the biochemical compounds in the strawberries happens. And there is a factors of the freeze drying that during the freeze drying and removing of the moisture content inside. So so there is a change over time.
So when you dried the frozen berries, did you analyze immediately, or was there further lag, and was that lag controlled or was that random?
When I do the freeze dry, right? So I do the analysis in the next day.
The next day. Yeah,
Yes.
Great.
And
For everything.
for everything.
Right.
And the strawberries are kept in the desiccant. And covered with the foil. Yes.
Excellent. Any more questions? All right, if there's no more questions, thank you very much.
I have frozen blueberries every morning. Yeah. Yeah. Blackberries.
Oh, I didn't even see it. Yeah, okay. All right, good. So last but not least, I'd like to introduce you to Ms. Saemi, and she's working on a project called Value Creation in the Dairy Industry in Regional Communities. Saemi, take it away.
Hello everyone, my name is Saemi Ito, and this project is supervised by Dr. Alex and Dr. Harsheen Kaur. Vietnam is one of the fastest growing emerging countries in Asia and an important partner of Australia. And with economic growth, the beef consumption is increasing. This bar graph shows the consumption amount until 2022 and expected amount from 2023. It has temporarily declined after COVID, but it's on upward trend. And import is also the same trend. But this red line is production. It's also increasing, but the pace is slower than consumption and import. But why does this happen? This pie chart shows cattle type in 2022 with 95% of beef cattle. And within beef cattle, there is 93% of farmers who have less than 5 beef cattle. So this means that most of the farmers are smallholder farmers who are not commercialized. So production does not keep up with the consumption rate. Based on this situation, there is an ongoing project funded by Australian Centre for International Agricultural Research, or ASIAR, to integrate smallholder into commercial beef supply chains. Purposes are strengthening partnerships with smallholder farmers and cooperatives, and improving supply efficiency and farmers' incomes. However, there are some limitations, such as low feed quality and productivity and poor connections and information exchange. And this can reduce the effectiveness of the project. And also there are some past projects, but these are in specific region, so there are some more region to explore. To fill this gap, I have set 2 project aims. First one is to identify feed nutrient availability and linkage in the value chain to improve future feed value chain systems and understand value of feed to farmers and motivation skills to introduce improved systems in the future. So this project is a preliminary study for future steps. Research questions are: how does similarity affect the quality and quantity? How can the further production involve central cost? What barriers impact buyers and farmers' access to grasses, alternative supplements, and other feed? What strategies are used by livestock producers to align fluctuating demands of feed with supply? The research area is Lowe, Quay Hop, and Tai Hoa district in Nghệ An province. And this is a newish place to research. And for research methods, baseline study and rapid value chain analysis were selected. And there are 4 steps. Discussion and interview were held for engaging the chain and understanding the market. Based on the interview, chain mapping are created, and thematic analysis and SWOT analysis are for identifying challenges and opportunities. So for discussion and interview, Focus group discussion were held in July, but I'd like to talk about interview, which I participated in. Period was 5th to 9th of September, and my interviewees are 15 livestock farmers and 10 forage farmers. Livestock farmers have 5 to 20 or 50 to 150 cattle, and they also have their forage and with less than 1 hectare, and age range is 30 to 50, and there is 10%, less than 10% of female. And for forage farmers, they also own their cattle for their property, 15, and forage area is around 1 hectare or 2 to 10 hectare, and age range is 20 to 70, and there is 20% of female interviewees. And for sampling, a regular used livelihood method or role is counted as one. This means that if there is one farmer who sells beef cattle and also sell their forage, they counted as one beef cattle farmer and one forage farmer because they have 2 roles. And there are 15 samples per 10 is enough for rapid variation analysis because it's preliminary study, and also information is important than quantity. And to gather the interviewees, local government asked farmers to participate in the interview. So interview structure is semi-structured. That is, information from conversation is also included in the set questionnaire like this form. And there are 2 questionnaire forms for beef cattle farmers and forage farmers and traders. And this photo shows how interview was done. And based on the interview, I created chain mapping to identify main inputs and check efficiency and effectiveness. In the interview questions, there is a question about before and after the chain, such as who did you buy the feed from, or who did you sell the feed to? And 2010 was beef cattle farmer because they own their forage, and remaining amount will be purchased from forage farmers. And forage farmers will purchase the seeds from input suppliers, and this is direct sales. And sometimes forage farmers rely on traders or collectors to collect and sell their feed to beef cattle farmers. So in this chain, consumers are beef cattle farmers and suppliers are forage farmers. And for analysis method, I'd like to talk about thematic analysis. Thematic analysis is commonly used for qualitative data analysis in wide range of fields, and there are some steps. Firstly, familiarizing yourself with the data and generating initial codes. This means that extract important and relevant information from the original responses from farmers. And categorize, put a theme and categorize code for each theme, and theme could be more than 2. And reviewing the theme and producing the report. I'm using Excel to do this analysis and put theme and sub-theme for each or interviewee's responses. And for result of thematic analysis, I'd like to introduce 2 topics, which is about instability of rice straw and unstable beef and buffalo industry to answer these research questions, which is about seasonality, barriers, and strategies. So rice straw is used all year round, but in winter, it's also used for kind of cushion for livestock to mitigate the cold weather. And it's also used for mushroom production and winter and spring production. And sorry for these long sentences, but this is actually the data from for qualitative data analysis. And so please check the highlighted keywords. And this purple speaking bubble is come from literature. And it also said that mushrooms or biochars using straw is increasing to reduce the straw burning. So there is off-season competition, which is related to research question 1, seasonality. Secondly, to make rolled straw, it needs to be sunny and dry weather, but the weather during the harvest season was bad, and sometimes rolled straw has more. That's harvest season is April, August, and November, and rainy season is May to October. So harvest season is at or close to the rainy season. So quality decline during harvest and rainy season has occurred. Next week, rice straw is sometimes in short in supply, especially in dry season like March, and prices increase. And according to NOC, rice straw is an important byproduct, especially in dry season. So off-season shortage and price increase is occurred, which is related to seasonality and barriers. And in terms of the second topic of unstable beef and buffalo industry, in spite of the increased trend of beef cattle in Vietnam as a whole, farmers fear that beef meat consumption is decreasing and value is low and unstable. A possible cause of this an oversupply of cattle due to lack of feed, but more research is needed to decide the cause. So there is a possibility of a negative reaction of feed supply decline and unstable beef cattle industry. Now, for research question 3, strategy. What kind of strategy farmers use to cope with the fluctuating feed supply? First one is multi-species forage and silage. So farmers own a variety of forage on their farm, and if they buy too much of feed, they make silage. However, byproducts from small farms can vary in quality and can reduce quality of their cattle. So there is still some room to explore quality testing or workshop. Second strategy is connecting producers and consumers by social media. So forage farmers and traders use social networking such as Facebook to sell their forage to beef cattle farmers. And from the literature, using online tools is a good way to strengthen community connections and negotiation power. In conclusion, there are some challenges and strategies to answer the 3 research questions. And recommendations include strengthening chain links and developing potential interventions through participatory workshops and investigation the cause of instability in the beef industry is recommended. And over the next month, I will conduct with SWOT analysis to analyze internal and external factors for challenges and strategies. Finally, I have been helped by many people. So I'd like to take this opportunity to express my appreciation to them. This is it. Thank you very much.
Thank you very much. Questions, please. Any questions? Yes, Rob was first, sorry.
Just beat you. Thanks. I found that really interesting, and I have a lot of. I'll think about a lot of things that I learned from the presentation. Actually, all the presentations today. My question is around methodology, and so obviously there's a lot of qualitative methodology being used here. What opportunities are there for you to use some of the new tools like AI when you are working in qualitative analysis?
So I was doing this manually, but if there is a tool called NVivo, which is help the categorize and put data, categorize and thing and yeah, so it's the tool for helping categorize the data, but it still need to consider by themselves. So, this method is kind of subjective rather than objective. And so you need to be careful to use these tools.
Next question, Arida.
It's a very excellent talk. Thank you so much, Saemi. I just wonder that the structure of Vietnam beef production system is smallholders. What is the aim of ACR? Are they trying to make production more efficient, or they're trying to suggest that the small farmers need to increase the capacity? Like maybe the system is the system as it is, and it's efficient for them because of more high risk, obviously, for the smallholder to have more beef. So I'm just wondering, what is the aim of this overall ACR product, ACR project?
So their main purpose is to include smallholders into commercialized system. So these smallholder farmers, they just own their cattle by themselves.
Mm-hmm.
But the government wants to increase the production, and so increase the income for smallholder farmers. So they construct the capacity building for smallholder farmers to commercialize. To commercialize.
I understand your answer, and I think it's a good one. But what I'm saying is that I think the risk is also the smallholders if something goes wrong. So even though the government is trying to encourage the smallholders to have increased capacity, what is the government doing to trying to help support them? Because at the moment it seems to be that they're not offering any subsidies, they're not providing financial aid. This is just where I'm coming from. Yeah. It's a difficult, complex question, but I just feel that there's more to the story.
Saemi, is the government doing anything to support the farmers?
To support the farmers?
Yes, anything. In this, with this issue, is the government, Vietnamese government, doing anything to support the farmers with this topic? If you don't know, that's fine.
So sorry, I can't get the.
So the government, the Vietnamese government, is it providing money, subsidies to the farmers? Is it providing advice? Is the government helping the farmers in any way with this topic?
Vietnamese government.
Yes.
So Vietnamese government in their plan. They I forgot what I want to say.
It's worthwhile exploring whether the government, Vietnamese government, are encouraging this by funding ACR, whether they would also be supporting the smallholders themselves in some way. What worth checking into, that's all.
So maybe take it offline and have a discussion. AP.
Semi, good presentation. I wanted to find out from you if you have an idea of the percentage of the people you interviewed that influenced the conclusions you came with, because you didn't tell us if it was 50% or 70% of the farmers that said what they said and made it to come up with the conclusion for each of the research questions you were trying to answer.
I didn't even I didn't understand that question.
Maybe I didn't also understand your methodology very well, but my question is, you had a number of people you interviewed and there were responses from them.
Yes.
And it was based on the responses that you concluded or came up with recommendations for each of the research questions. Yeah.
Recommendation from research question.
If I may. So you interviewed, let's say, 10 farmers.
So what percentage of those 10 farmers or 17 and 14 farmers you interviewed, what percentage of them gave similar responses that resulted to your conclusion?
Yeah.
Was it 70% of 14 of the farmers or 50% of 14 of the farmers? Do you have an idea, because you didn't present us? I wanted to know.
Very depend on the answers and the responses and the question. And I didn't count the percentage of each because it's a preliminary study, so and number is only 15 or 10. So statistically it's very important. And if it's for bigger research, answer it's counting until answer is saturated. Same answers will come.
Yeah.
So for this research, sorry, I didn't count this one.
So Saemi, you reached a conclusion with your results, correct?
Yes.
Okay, so you reached a conclusion. Out of those 15 farmers, how many of those 15 farmers agreed with your conclusion in your survey? And if you don't know, just say to Ave, I don't know. I'll answer later. I'll find out and I'll get back to you. Maybe just say that if you don't know.
So yes, I don't know, but
yeah.
Maybe it's not the focus point for this research.
So,
there's an online question.
The question is, I was interested in the low participation of women in livestock production. Has any thought been given to family production unit approaches, including women and youth?
Also, not focus point for this research, and the ASEA project is also focused on the gender differences, but for research, I didn't analyze this point.
So Saemi did not analyze this point, or she didn't address that point at all. All right, any more questions? Another one? Yes.
Information update more than anything. It says ACIAR has been focusing on supporting the smallholders in agriculture in developing countries like Vietnam. The economic integration globally and the economic development of the country has highly likely marginalised small-scale production. That's where UTAS has been leading 4 different ACIR-funded projects in beef cattle industry in Vietnam. In the last 10 years, we've been helping the local communities in remote areas to overcome technical and marketing constraints to establish the domestic beef cattle supply chain.
Sounds like
Just for some history, I thought that might be important.
Right, all right, well, thank you, Saemi, thank you very much. Just stay here. I thought it fitting, since we have Saemi up here, that I would, somebody asked me to recite Japanese poetry, so this is a Japanese haiku. Japanese poetry. It's about Box Hill Station in Melbourne. It has a red engine tucker box. So this is very short. Springtime, Box Hill Station. Commuters rushing to their destination. Red engine going nowhere. Thank you. Thank you, Saemi. Thank you, everybody. I've had a wonderful day. Thank you so much.
Thanks,
Thanks, everyone. That have done some, yes, Rob.
Can you share the QR code again, please?
Yes, the QR code open. Can we have the QR code? Thanks.
Okay, let me see.
I clicked through and submitted all zeros, so I better update.
Good job, man.
Thank you very much, everybody.
I know. I hope it doesn't affect the average.
Thank you so